All right, everybody, ladies and gentlemen, colleagues, distinguished guests, everyone joining us in person here in Cleveland and virtually, welcome to all of you. I'm Jeff Bennett.
And I'm Abigail Kacpura. We're the directors for this event, and we are very honored to stand before you to kick off Cleveland Clinic's 10th annual Genetics Education Symposium. This is an event that brings together the brightest minds in the field, unites those who are passionate about unraveling the mysteries of our genetic composition, and those who are committed to pushing the boundaries of medical knowledge and practice.
Today we'll be reviewing the latest advances in the genetics and genomics of cardiovascular conditions and provide state-of-the-art information to healthcare providers, trainees, and students. Our goal is to establish connection and collaboration between the fields of genetics and cardiology, fostering mutual interest and collaboration, all in the interest of advancing science and patient care. Over the next few hours, we'll have the opportunity to learn from distinguished faculty of leading experts in the field, and they will guide us through a wide range of topics, from fundamentals of genetics to the latest advancements in the care for genetic heart disease.
We are confident that you will leave this course with a deeper understanding of the interplay of cardiology and genetics, and with renewed enthusiasm for your work. Our hope is that the knowledge and skills you gain here will help you make a real difference in the lives of your patients. In conclusion, I would like to express gratitude to all of the participants for joining us in a commitment to lifelong learning and improving patient care. I would like to thank our esteemed faculty for their invaluable contributions to this course. Next up, I have just a few housekeeping items before we get started. So many thanks to our exhibitors in support of this year's symposium. We wouldn't be able to do it without you, and we encourage you to visit their tables during breaks and lunch. Our gold level exhibitors include Rocket and Caniska Pharmaceuticals. At silver level, we have Sanofi, and at the bronze level, we thank Alnylam, Amicus, AstraZeneca, Bristol Myers Squibb, PSC, the DDC Clinic, Ionis Pharmaceuticals, Pharming Healthcare, and Prevention Genetics. I know we just had some small bites, but for those of you already thinking about lunch, it will be fantastic and will be served in the ballroom on the second floor. PDFs of the faculty presentations will be uploaded throughout the course. Please see the online instruction program book for instructions. And then for those of you seeking NSGC credit, please note that in-person registrants must sign in and out at the registration desk. I know many of you have already signed in. Live stream attendance will be tracked via WebEx. And all participants must provide their NSGC user ID when completing the required course evaluation in MyCME. Detailed instructions will be provided to you after the course. You will be notified by email when claiming credit is available, and please see CME staff at the registration desk with any questions. For questions and answers after each lecture, please press and hold the button on the microphone at your seat before speaking. All right, without further ado, we're going to get started with our first speaker today, Joseph Liu. He is one of our own here at the Cleveland Clinic. He is a genetic counselor, and he earned his undergraduate and graduate degrees at Case Western Reserve University. He's a licensed genetic counselor in Ohio, Indiana, and Florida, and is a diplomat of the American Board of Genetic Counseling. He is fantastically knowledgeable in the field of cardiogenetics, and he was starting us off with our first talk of the day, a genetics primer, to go over the basics of clinical genetics and genetic testing.
Hello. Oh. All right, well, thanks for that introduction. Again, my name's Joe, and I have the challenging task of, in just 10 to 15 minutes, priming us for the exciting information we're going to be hearing from our faculty today, and hopefully priming us, I think, to take something back to our clinical practice. Let me see here. So do I click with this? Okay. I have no disclosures. So broadly, cardiogenetics, refers to, in general, 5 domains, 5 disease groups, shown here, starting with hereditary cardiomyopathies, oftentimes referring to hypertrophic cardiomyopathy, dilated cardiomyopathy, arrhythmogenic cardiomyopathies, and restrictive cardiomyopathies. There are also the inherited arrhythmias, which oftentimes are related to genes involved in the ion channels. Most commonly, the big three being long QT syndrome, Brugada syndrome, and catecholaminergic polymorphic ventricular tachycardia. There are the inherited aneurysm disorders or arteriopathies, most of which include genetic aortopathies, some of which involves connective tissue disorders. There are the non-syndromic and syndromic causes of congenital heart disease. And then out here on an island are the inherited dyslipidemias like familial hypercholesterolemia, cytosterolemia, and a few others. And I intentionally overlap some of these categories because even though the genetic basis of these conditions may be distinct, the presentation and the phenotype that we see oftentimes overlaps. Now, there's a saying in clinical genetics, if you have a medical genetics background, that rare diseases are like zebras. But I think in cardiogenetics, it's probably better to describe some of these conditions as just different kinds of horses. Because in general, inherited cardiovascular disease is incredibly common in the general population. Now, if you look at data from registries and even population-based studies looking at the prevalence of these phenotypes, starting with congenital heart disease, about 1 in 100 newborns are born with congenital heart disease. Familial hypercholesterolemia is prevalent in about 1 in 300 individuals. Hypertrophic cardiomyopathy is prevalent in about 1 in 500 individuals, and so on down this list. But what we know, even looking at from a genotypic perspective, meaning from the perspective of genetic testing in the general population, oftentimes in healthy adult cohorts like the UK Biobank, that the frequency of genetic variants which are associated with inherited cardiovascular disease is probably actually higher than what we phenotypically see. And some of the numbers here are reflected. And even from the perspective of what we would oftentimes consider rare diseases like Marfan syndrome, the frequency of likely pathogenic or pathogenic variants in the UK Biobank has been estimated to be close to 1 in 3,000. And so this is reflective both of the variability in how these conditions present, but also the fact that there is what we call reduced penetrance, meaning that not everyone with the genetic predisposition to disease, in fact, shows clinical evidence of disease. And so part of the reason that I think cardiovascular genetics is so interesting is that the architecture of what genetically, how genetic variation causes cardiovascular disease can be complex. Most of what we'll be talking about today is monogenic, is considered monogenic or Mendelian disease. It's the type of condition that you would think about where there is one clinical entity caused by genetic variants in one gene. And these tend to be variants or genetic differences that have a very low frequency in the general population. And because they're very rare, and really the opposite of that, because they have a high degree of risk associated with them, they're very rare in the population. And typically with monogenic disease, a variant associated with monogenic disease has a high enough risk that it probably gets you very close, if not to the threshold, for actually manifesting signs of that disease. On the far other end of the spectrum, here in the green box, you can see that there are diseases which are polygenic in nature, meaning that rather than being one very rare genetic variant associated with a high degree of risk, we can see a multitude of common genetic variants in the population, each of which may only have a very small effect size or a very small degree of risk. But if you add together 20, 30 of those common variants, you can have someone who, again, develops clinical evidence of disease. And then in the middle, depending on how you conceptualize oligogenic disease, those are conditions that are somewhere in the middle, where you have variants that are uncommon and have a moderate level of risk associated with them, but generally no single variant by itself is going to be responsible or able to cause clinical disease. And of course, we have to add in environmental factors as well. Now, a few examples in terms of what the inherited cardiovascular disorders that can, or cardiovascular disease in general, that can present as more monogenic versus polygenic. Long QT syndrome is a great example of a inherited cardiovascular disorder that is strongly monogenic, and it's evidenced by the fact that most individuals who meet clinical criteria for long QT syndrome have an abnormal genetic test. But we do know that certainly from more recent studies and research done in acquired long QT syndrome, that there is also common genetic variation that plays a role in the length of the QT interval. Conversely, coronary artery disease is something that is most often multifactorial. It's a combination of both environmental factors and, in most cases, polygenic risk, but obviously there is still a monogenic component to that in the shape of familial hypercholesterolemia. Again, we're going to be focusing most of our speakers today on monogenic disease, and it's unique because there's a very tight correlation in monogenic disease between the phenotype, the clinical entity, and the genotype, the specific genetic variant or mutation that's responsible for disease. And it's a relationship that's really one of causation. And when it comes to evaluation of cardiovascular genetics, we always have to start with the phenotype. And that's really where, today, the name of today's symposium is multidisciplinary care. This is really the stage where, it's most, in some ways, it's most crucial to come together. The cardiovascular testing, the echocardiogram, the imaging, the EKG, all those things are important in understanding what we're even supposed to be looking for. Medical geneticists then can contribute a piece of the dysmorphology exam in inherited cardiovascular conditions where there are extra cardiac features. And genetic counselors can also lend their expertise in understanding how it's manifesting in the family history. All those constitute what we consider the phenotype. And that can give us a clue in terms of genetic testing to understand what should we even be looking for in the genotype. However, I included this picture of the iceberg because most icebergs, as this picture shows, are hidden under the surface. And even though a phenotype may give you a clue as to what's going on, sometimes it requires us to understand the gene involved, the variant involved, the effect of that variant on protein function, and even the domain in which the variant is located can then go back and inform to us what to do with the phenotype and how to manage it. And I think today we'll have a great opportunity to learn more about that from our other faculty. Now, how do we look below the surface? What kinds of genetic tests might we expect to come across in cardiovascular genetics? So these, I only touched on 3. There are a whole bunch. And for the sake of time, I'm just going to stick to these and just highlight some of the differences. And there's going to be 3 I'm listing here, and I'm actually sorting them and going in order of the highest level of resolution to the lowest level of resolution, starting with the karyotype. Karyotype analysis, a commonly known test done for things like trisomy 21 or Down syndrome or Turner syndrome. It's able to detect gains or losses in whole chromosomes as well as structural rearrangements, and it does not select for a specific chromosome. It checks all the chromosomes. Going to the next level will be the chromosomal microarray, which is able to detect gains or losses in genetic material that are smaller than the size of a whole chromosome arm. Usually it's within the range of between 20 to 50 kilobases, although in clinically relevant regions, the resolution can actually be higher. You may have heard of microdeletions or microduplications, such as 22q11.2 deletion syndrome. That is an example of a copy number variant, and it again would check across the whole, across every chromosome. And then a lot of what I suspect we'll be talking about today is something called sequencing, oftentimes referred to, and specifically a type of sequencing that probably we'll be talking the most about today is something called short read sequencing or next generation sequencing. And essentially what this type of test can do is it actually gets down to the level of the single nucleotide. And so variants that are essentially a single nucleotide substitution or even small insertions or deletions are really only identifiable by sequencing. The advantage also of sequencing is you can go as focused or as broad as you would like. You can start with a few select genes, which we would call a panel. You can then go even larger to all the protein coding regions, which are essentially all the genes, which we call the exome. And then you can go beyond that to not only include all the genes, but even the parts of the DNA that only make it to RNA and never become a protein, the non-coding regions, which would be something we call genome sequencing. Fundamentally, all of these can achieve the similar level of resolution, although the different sequencing tests can vary depending on how much they're able to detect things like copy number variants, structural rearrangements, and other types of more eccentric genetic variation. So now that we know what tools we have in the genetic toolbox, how do we know where to look? Well, this really comes down to the idea of gene curation, understanding which genes are associated with disease. And there is a collaborative group called the Clinical Genome Resource, which is a collaborative essentially assembly of experts in genetics that have come up with a criteria for how we determine what genes are associated with cardiovascular disease and genetic disease in general. And I'm not going to go through the details here, but essentially both real-world clinical evidence, which is here described as genetic evidence, for example, a family with 8 individuals with the same clinical phenotype that have the same rare variant in a single gene, that data can be used to support that a given gene is associated with disease. Experimental evidence, on the other hand, is largely stuff done in a lab, things like models, which demonstrate that gene function in some way or disruption of gene function results in a specific phenotype. And what we end up with is really a number of categories that you can receive for a specific gene. So some genes that you may read about or hear about are candidate genes. These are typically referring to genes that have limited evidence. And it's really only the genes that have moderate, strong, or definitive evidence that are most likely to be available and useful from a clinical perspective, because these have the highest level of evidence that variants in these genes can be associated with disease. If there is contradictory evidence, genes can also be considered disputed or refuted. Once we know where we're looking in the genetic code, we have to understand what we're looking at, and that's variant interpretation. And this is a very large table from the 2015 guidelines from the American College of Medical Genetics and the Association of Molecular Pathology, specifically for those sequence variants. And you don't have to know the details, but it's really important to know that there are multiple lines of evidence that are required to determine whether a variant is associated with disease or not. And in fact, even after you look at all these rows of information, how common it is in the population, the computational prediction, functional data, clinical data from a family, inheritance information, you may still not arrive at a conclusive decision as to what a variant means with respect to disease. In general, once you look at all the data, the variants can often be classified either into likely pathogenic or pathogenic, which are your positive results. Those are variants that have over a 90% probability of being disease-causing. You can have variants that are likely benign or benign, which are over a 90% likelihood of being harmless. And then you have everything in the middle, which we affectionately call in genetics variants of uncertain or variants of unknown significance, sometimes referred to as VUSs. But these things change over time. And just to illustrate this, the picture on the left is the 2019 ClinGen group that initially curated the hypertrophic cardiomyopathy genes. At the time, they listed 11 moderate, strong, and definite evidence genes that were known to be associated with HCM. And just in August of this year, they published a preprint for the reappraisal of the HCM related genes, and they're now at 29. So the information is changing rapidly in cardiovascular genetics, as is our understanding of the variants that we're finding. And so this graph at the bottom shows data from a commercial laboratory, which tested over a period of 8 years. They tested over 1.6 million individuals, many of whom had cardiovascular genetic testing. You can see that about 9% of the VUSs or variants of uncertain significance that were identified were reclassified. Majority of them ended up being downgraded towards benign, but about 2% of all VUSs were upgraded to pathogenic as new information was learned. And so I'm very excited to end my talk because we're going to talk more about where we're going and talk a bit about what we've learned in the recent years with our other speakers. I'm not going to get too much into this, but I think that there's a lot on the horizon in cardiovascular genetics. Thank you.
Thanks, Joe. That was a fantastic talk that gave us a great overview, but also touched on the complexity that we have in genetics. All right, next up, we're going to get deeper into the details. So for our next speaker, I would like to introduce Vidu Garg. He's the director of the Center for Cardiovascular Research and the Nationwide Foundation Endowed Chair in Cardiovascular Research at Nationwide Children's Hospital in Columbus, Ohio. He is a professor in the Department of Pediatrics and holds an adjunct appointment in the Department of Molecular Genetics at The Ohio State University. He also serves as the director of translational research, and is a board-certified practicing pediatric cardiology in the Heart Center at Nationwide Children's Hospital. He completed his pediatric residency at Nationwide Children's Hospital in 1997, after which he spent 5 years at UT Southwestern completing his pediatric cardiology training and a postdoc research fellowship in cardiac development and cardiovascular genetics. Subsequently, he joined the faculty at the University of Texas Southwestern before returning to Nationwide Children's. Dr. Garg's research laboratory focuses on molecular pathways controlling cardiac development and the genetics of congenital heart disease. Dr. Garg will be speaking to us about genetic contributors to bicuspid aortic valve and left-sided obstructive heart malformations.
Thank you, and thanks for the very kind introduction, and thanks, Jeff and Abby, for the invitation to speak to you today. So, as was mentioned, I'm a pediatric cardiologist, a little bit of fish out of water here, and so I do a lot more cardiac developmental research. So I'm going to start off a little bit. We had a really nice introduction from a genetics standpoint, and so I'm going to give a little bit of an introduction from a of congenital heart disease. And so, as Joe mentioned, it's the most common type of birth defect with an incidence of about 1 in 100 live births. And I just want to mention here that excludes bicuspid aortic valve, which is one of the topics, which in its of itself has a prevalence of 1%. And I think it does complicate some of the genetics as we try to study this. We now have a disease incidence of about 2% in the population. It's a leading cause of infant mortality due to birth defects. I think the other thing is that it encompasses just a spectrum of malformations. Something that's really simple, if you think of just septal defects, like an atrial septal defect or ventricular septal defect, something more complex where you have the whole left side of the heart being hypoplastic left heart syndrome. We've seen a lot of improvements in our medical and surgical management of these patients over the past 3 decades, with a growing population of congenital heart disease survivors, which again raises the question of really understanding why these diseases occur and the genetic contributions. And we've also seen many of these survivors suffer from long-term morbidity and mortality, specifically from cardiac and non-cardiac complications. And epidemiologic studies from the 80s really suggested a role for genetic contributors. And so with these advances that we heard about in terms of genetic testing, we've identified chromosomal abnormalities, copy number variations, single genes that really are contributing to congenital heart disease. But still, the majority of these cases are unknown. And one group of those that I think we still don't really have a good handle on in terms of congenital heart, the genetic etiologies, is this bicuspid aortic valve and left-sided obstructive heart malformations. I'm going to either call them left-sided congenital heart disease or left-sided obstructive lesions. It's a long bunch of words. But essentially, I wanted to kind of just briefly define what these are. I know there's a broad audience. So these are defects that are really affecting the left side of the heart. So it can be anything affecting the valve. So aortic valve stenosis, anything affecting the arch, and so that'd be coarctation of the aorta, or inside the heart. So mitral valve stenosis, or again, hypoplastic left heart syndrome, where there's a variety of structures on the left side that are small, or Schoen's complex, which again is a variety of defects that affect the mitral valve, the subvalvular aortic area, and also coarctation of the aorta. I am not going to discuss other lesions which are also considered left-sided obstructive heart malformations, such as supravalvular AS, which I think we'll hear about in the next talk, interrupted aortic arch, which is kind of linked more to DiGeorge syndrome or 2.2 syndrome, or other mitral valve overlaps. So when you think of these lesions, they can be associated with syndromic disease. I would say the 2 most common would be Turner syndrome and Jacobson syndrome, where you more commonly see left-sided disease, but there's a variety of chromosomal abnormalities or syndromic disease that you can find a bicuspid aortic valve or some types of these. And that's really not going to be the focus of my talk. I really want to look at non-syndromic congenital heart disease. And the reason for that is we've known for decades that there's familial clustering with bicuspid aortic valve and other left-sided disease. Early as the 1970s, I think there's 30 consecutive patients reported in JACC where they essentially found 10% of the relatives had bicuspid aortic valve. This is a large meta-analysis that just came out in the European Heart Journal. Again, very similar recurrent findings in terms of if you look among first-degree urologists by bicuspid valve, you're going to find 7 to 9% of either having VAV or associated aortic dilation individuals. And the same goes for when you go to other left-sided congenital heart lesions. This is a paper from the Netherlands in 2011 where they looked at 142 consecutive patients and screened first-degree relatives. And again, 8% incidence of congenital heart disease in a first-degree relative, bicuspid valve being most common. This is a picture, again, for this audience. This is from Woody Benson's work from years ago now, but these are the pedigrees that we see when we see patients with bicuspid valve. They commonly have somebody else with bicuspid valve, somebody else with high plus left heart syndrome. And if you look amongst population-wise, we're getting about 18% in about half of the families. You can find someone else with congenital heart disease. So when you see this, it kind of led us to think, well, there must be common genetic etiologies of bicuspid valve, coarctation, and high plus left heart syndrome. And so as Abby mentioned, I also do development. And so when you go back to cardiac development, and this is just kind of a depiction over the first 8 weeks of gestation, you start out with the heart, in around 2 weeks as a cardiac crescent structure, ultimately fuses into a linear heart tube and starts beating, undergoes white-bird looping, and by 8 weeks of gestation, you essentially have a 4-chambered heart with a separate aorta and pulmonary artery. We've learned a lot about heart development over the past 2 to 3 decades, and now we can draw these really nice fancy pictures in terms of all of the different cardiac progenitors that are important for forming the heart, from the first and second heart field that are going to essentially, the first heart field will form the left ventricle, the second heart field will primarily form the right ventricle and the outflow tract, other lineages that are contributing from the endocardial cells that will contribute to the cushions, both the atrioventricular and the outflow tract cushions, neural crest cells that will come in also to the outflow tract, and even epicardial cells that are reforming the coronary arteries and mouse models contributing to the atrioventricular valves. So when you see all of these structures and then you think about this forming heart, that this is the left side of the heart, with the ventricle, the outflow tract, arch artery, I can now think of all these different cell populations that are contributing to it. It becomes a little bit difficult to think that there's one gene in one cell population that's causing this spectrum. And so maybe it was a little naive, I think, when we saw those pedigrees, that there was a single gene that was essentially causing this. And I think now, going back to Joseph's slide here, we were thinking more Mendelian, and there still may be some that are truly Mendelian cases, but there's likely more of a susceptibility alleles and oligogenic phenomena, likely with environmental factors that are ultimately leading to this. And when you look among those families, there is reduced penetrance and variable expressivity that may be contributing to some of the phenotypes that we look through in these families. I pulled this. I'm not going to go through all of this, but this is a really nice review from Andrew Landstrom in 2021, looking at the genetics of VAV. I think the main thing take home here is of VAV and other left-sided diseases. Here's bicuspid valve, aortic stenosis, high plus left heart syndrome, and coarctation of the aorta. There's many genes that have been implicated. I'm going to talk a lot about Notch1. It's a gene that's my favorite, so I will go into that into most detail today. And it is kind of in the middle of all of these. The other thing that I really want to point out, a lot of these have aspects, which means that there's just not a lot of robust evidence of association. It's been described and the papers are very well done, but it's just the repetition as we're getting better and better in sequencing more. I think some of these asterisks will be removed with time. So I'll talk about a few of these genes, mainly the ones that are without the asterisks. This is where I got started now 20 years ago, essentially finding some of these families. This is a family I found with bacterial bacteria at UT Southwestern about 20 years ago. It's a family from Paul Grossfeld at UC San Diego, who's also a pediatric cardiologist. And again, when looking amongst these families, the primary phenotype we really saw was bicuspid valve and its associated diseases in terms of aortic dilation and calcification. I will note that there are people with tetralogy of Fallot, mitral valve disease, a hypoplastic left ventricle, variant with double ventricle microatresia amongst these families. We were lucky enough, even when we submitted for a grant, we were told we were never going to find this gene, but we were lucky enough to identify a gene that was causative. It was Notch1, and essentially it's segregated with all the affected family members and usually lost patients in both of these families. So Notch1, it encodes a transmembrane receptor, functions in a very highly conserved intracellular signaling pathway, which is shown here. Basically, two families of ligands, the jagged and delta family of ligands. Once you get binding to the receptor, there's a series of cleavages that Notch1 undergoes, and it ultimately translocates to nucleases and functions as a transcription factor. Notch1 was known to be important for kind of vascular development, knock it out in mice, it leads to a variety of vascular defects. Important for us, it was highly expressed in the developing allotract and the aortic valve in mouse models. So as a phenotype was primarily bicuspid valve, that's what we focused on. And so as I mentioned, the AB has a prevalence of about 1%. It's fusion of two of the leaflets that we commonly see. And patients, essentially, this is a child that I follow, will have progressive aortic valve stenosis and valve stenotic early. Some patients are completely asymptomatic with BAV adulthood, but when they become adult, they can develop ascending aortic aneurysms and calcific disease. And so we've used this as an entry point to study all of these diseases. I'm really only going to talk about congenital heart disease in this talk, but we have done work making Notch1 in aortic aneurysms and a lot of work looking at Notch1 in calcific disease. Some of our early work really tried to model it. When we look at Notch1 heterozygous mice, there was no phenotype. It was very lowly penetrant. I think you can barely see here, but about 10% or less than 10% of mice will have a bicuspid valve. But some of our work with calcific disease led us to generate compound mutant mice, which were mutations in Notch1 and also nitric oxide synthase 3. And when we got these, we were very excited. There were some adult survivors, and they were very highly penetrant bicuspid valve phenotype, along with aortic dilation. But I mentioned only about a third of these mice lived. When we look at the embryonic phenotypes, it's very interesting. We actually ended up seeing a kind of more of a coronal-truncal or tetralogy of below phenotype, where we saw like ventricular septal defects, no overriding aorta that's pictured here, and pulmonary valve stenosis. Again, going back to that original family that we actually did have some of the tetralogy of below. Subsequent work by the Wessel's group and then our evening really has linked Notch1 mutation to right-sided cardiac anomaly. I really like this paper from the Wessel's group. They looked at 428 probands for Notch1 mutations. They found 14, which just gives you an incidence of kind of how often you're going to see this. But the majority of them were in familial left-sided congenital heart disease, and it's about 7% of those families, and about 3, they had, so 1% incidence in sporadic left-sided congenital heart disease. And when you looked at those 9 families, there was another family member who had right-sided congenital heart disease. And so it becomes really more of a conal truncal heart defect as opposed to in the original paper where we went to bicuspid valve. And this is similar work from Bernard Keaveney, where he looked at 829 individuals in tetralogy, and about nearly 5% had predicted pathogenic mutations in Notch1. So this has been expanded. Some early work from Tim McBride, looking at Notch1 in a variety of left-sided congenital heart defects. A lot of these were inherited missense variations, but then there's other work from Tim Olson's group, this is Matt Durbin at Bennett now and Kaz Hong's group, and then even from the Pediatric Cardiac Genomics Consortium, where we're essentially identifying Notch1 variation. Either it's a missense inherited or de novo variation with a variety of left-sided congenital heart defects, kind of linking the VAB and the other left-sided congenital heart lesions with Notch1. This is just a little bit of development because it'll be relevant to some of the stuff I want to talk about. We were able in our mouse model to really show what was Notch1 in the endothelial lineage that was causing these phenotypes, including the ventricular septal defect and the tetralogy of below phenotypes, and not in the other lesions. And this is also shown by Bin Zhao's group at Albert Einstein and Jose de la Pampa's group. And specifically now we know that it's Notch1 after EMT in terms of valve remodeling that causes a bicuspid valve. And then Notch1 has just been studied by many individuals, and we now linked it to Adams-Oliver syndrome, which is also associated with pubis aplasia, digit abnormalities, vascular malformations. And I really like this recent paper by Stanley et al. That came out in just this year, where they looked at 11 families, 33 individuals with Notch1 pathogenic variation, and they're actually expanding the phenotype. And so you can see from 2005, there's been a significant phenotypic expansion of Notch1 pathogenic variation. It's not just BAV, but now tetralogy of Fallot, and then potentially other vascular anomalies associated with it. Last minutes of this, I'm not sure if I'm good on time here. But I will continue here. Okay. And some other genes that have been linked that we've been studying is GATA5. There's some pathogenic variation that was linked by Christopher Samsarian's group in Australia in 2012 that were linked to bicuspid valve. And we've done some work and others. It essentially was based off of this finding from Mona Newman's group, where you completely delete GATA5 in mice, you get about a 25 incidence in 25% incidence of bicuspid valve. Interesting work from her mouse models was that there was dysregulation of Notch and nitric oxide signaling in those models, which actually goes back to some of the earlier work that I presented, that these genes may actually be functioning together in terms of being important for valve development. We've done some work that I'll briefly touch upon where we now, if you think of these as susceptibility alleles, and we can't really create these phenotypes in the heterozygous state in mice, we created compound mutant mice with both Notch1 and GATA5. And these mice essentially have developed aortic valve stenosis. When you look in the mouse model, stenosis is progressive, kind of like that patient I mentioned. And then they have other phenotypes, as you can imagine, in terms of left ventricular hypertrophy that they'll develop with time as they age. About half of them have bicuspid valves with increased incidence of 25% included GATA5 null. They have aortopathy, as you would expect. And then when you look earlier, they're already having thickened aortic valve, which is that embryonic 18.5, 14.10. So we're very excited to start using this model to study congenital aortic valve stenosis. We've also started thinking about other genes in the Notch signaling pathway. So this is some work done by Jose de la Pampa, reported last year with MindBomb1. So this is a regulator of kind of ligand endocytosis, which is an important process of Notch signaling, where MindBomb is a ubiquitin protein ligase that's important for this process. They found mutations in MindBomb1 to be associated with bicuspid valve in humans. Similarly, there's a ubiquitinase that regulates MindBomb1, USP9X, that we found was important in terms of aortic valve calcification. We take it out in mice, we can get aortic valve stenosis. This gene is actually linked to more X-linked intellectual disability in humans, but there was a recent paper in a boy who had a USP9X mutation, and they actually had left-sided congenital heart disease with bicuspid valve importation. The idea from this is just an entry point. There's likely many of these genes in this pathway that can then function as modifiers to ultimately lead to left-sided congenital heart disease. Again, going through that complexity of disease. I'm just going to briefly touch upon some of these last kind of known genes. They're essentially genes that have been shown to be important in outflow tract development. So SMAD6 to regulate the BMP signaling pathway. It's highly expressed in the outflow tract. Now, 10 years ago, there's been rare variants in SMAD6 were linked to bicuspid valve and coarctation, subsequently reproduced. But as Joseph mentioned, a small percentage are going to be related to SMAD6 mutation. Similarly, ROBO4, another slip robo signaling, very important from a development standpoint to be important for outflow tract development. Some work from Hal Dietz, where they identified 2 families that had bicuspid valve and were aneurysms. Similarly, there's enrichment of predicted rare pathogenic variants in ROBO4 in this patient population. These capitulated in mouse models with bicuspid valve, aneurysms, all supporting kind of, if you look through the ACG criteria for some of these variations to be important in this disease process. Another gene that's been linked is myosin heavy chain 6. This has been linked to a variety of congenital heart diseases, initially with atrial septal defects. I think it's a very interesting gene when one thinks about its role in terms of causing left-sided disease or congenital heart disease. There's also a link to outcomes, which I think is where we're headed. We'll hear about some of the talks later today. But initially reported that this could actually impact how patients with single ventricle heart disease will do in terms of having reduced ventricular function later on in life. This has been recapitulated by multiple groups now, including the pediatric cardiac consortium. They mentioned myosin heavy chain 6 is linked to not only hypoglossal left heart syndrome, but other reasons such as ASD and orientation of the aorta. I think this is still in its early stages in terms of really trying to understand how this is actually causing either congenital heart disease or potentially linking it to outcomes. Some larger studies there. I'm going to end on this. If we actually do exome sequencing on those families that I mentioned from Woody Benson, so I did this with King McBride and Peter White and Nationwide Children's, what happened? So these are 19 cases that we did exome sequencing. I think you, in about 25%, so 5%, you'll get genes that we know, ROGO4, myosin heavy chain 6, MAC1, which are two that are also been implicated. In other cases, we get candidates, really good candidates. CASI1, ROC1, these are, CASI1 is important for myocardial development. SMID1 is important for myocardial development in animal models. BMP10 and ROC1 are for alpha tract development. We had to do functional studies in terms of really trying to link some of these genes. And so, if you do it clinically, are you going to get something to take back to the patient? Sometimes, but other times you find interesting candidates that are hard to demonstrate. So I think as we move forward, as we sequence more, I think we're going to expand this group and of contributing disease genes and really try to understand what role they're playing. And I think what I'm hopeful for is as a cardiologist, can we get to better genotype phenotype correlations in terms of really looking at outcomes and what are these players. So thank you. And hopefully I didn't ruin my time.
Any questions for Dr. Garg?
Yeah. I mean, I think if you have familial disease, it's probably reasonable. I mean, you'll get something that potentially that you could return back. I think if you're looking at an isolated sporadic, it can be discussed. I mean, from a pediatric cardiologist standpoint, we're going to be saying you need to screen your first degree family members. We're going to be saying if you're planning to have children, that we want you to do a fetal echo. So there's stuff that we're already implementing from a standpoint, from a clinical standpoint. The question becomes, what's the value of doing a genetic test on that patient? That's where it gets a little bit tricky. Knowing that we're likely to get, especially for a single. I mean, you saw that one for Notch 1, it's 1% or less than 1%. And then you're just taking all of these, and how many do we know, and how many are we going to get to that point that we can actually return that result? That's something that we're looking into right now. No, I mean, I'm interested in two things. That is one aspect, is looking at, for the bicuspid valve aortic dilation, is there something that we can study? And then the second is with this new model of aortic valve stenosis, is there ways that we could either remove continue the remodeling of that myxomatous valve in the neonate, or potentially prevent that progression once you see it? So I'm a little biased as a cardiologist. I prefer going broad because I think you can just see with Notch 1 as we've expanded. I think we have biases built in terms of where it was first discovered. It doesn't mean that they're not involved. If you go back from a development standpoint, a lot of these genes are playing a role in a variety of others. And so I would go broad. That's just my bias. It probably does. I mean, I'm still going to go a little bit in terms of if you look at some of these families that there's still some larger players that are driving that, as opposed to going into truly complex disease where it's multiple genes that are doing that. That's just my bias. But if you were to look at just all sporadic and doing, there may be a role. I don't think I can speak to that yet.
Excellent lecture, Dr. Garg. Next up, we will be hearing from Beth Kozel, also joining us from Nationwide Children's Hospital. She is Chief of the Division of Genetics and Genomic Medicine and the Director of Constitutional Genomics Translational Research in the Steve and Cindy Rasmussen Center for Genomic Medicine. She is also a principal investigator in the Institute for Genomic Medicine. She has extensive experience in genetics, vascular, and matrix biology with research focused on the study of two elastin insufficiency-related diseases, the neurodevelopmental condition known as Williams syndrome and familial supravalvular aortic stenosis. She has made key contributions to the field of connective tissue biology. More recently, her efforts have focused on designing methods for genetic modifier discovery in rare disease cohorts. She graduated summa cum laude from Washington University in St. Louis with a bachelor's degree in biochemistry in 1996. She attained a medical doctorate and doctorate in cell biology and physiology at the Washington University School of Medicine in 2004 through a combined MD-PhD program. She completed her residency in pediatrics at St. Louis Children's Hospital and her residency in clinical genetics at WUSM. She's board certified in pediatrics and clinical genetics, and her talk today is entitled Rare Meets Common, Expanding the Spectrum of Elastin-Mediated Disease.
Thank you for that. And thank you all for having me and for listening today. My name is Beth Kozel. I'm going to talk about a lot of the work that I did in the last 8 to 9 years when I was at the NIH. I just moved to Nationwide in the last few months, so I have to acknowledge that. But do want to sort of get you started by talking about my favorite gene to introduce things, which is elastin. So this is a picture of a cross-section of a blood vessel wall that has been autoclaved. And the autoclave kills all the cells. And so what you can see here in the black area is where the cells used to be. And withstanding that autoclave is the elastin. It is the connective tissue that sort of binds all of those cells together. So it comes out of the cell as a soluble monomer and then multiplies together to make this sort of chain of connective tissue that provides recoil when your tissues stretch. Name is elastin. It's involved in sort of providing that recoil to your blood vessels, to your lungs, to your skin. It is extraordinarily long-lived. So it actually gets made during a very short period of time during your infancy and into adolescence, and it has a half-life of 74 years once you put it in. And so to a certain extent, if you don't do it right the first time, you don't really get do-overs. And so a lot of what my lab and the work that we're doing is trying to understand the gene variation associated with that and seeing if we can find ways to intervene and sort of fix that problem. So most of the place where we see elastin insufficiency is associated with a microdeletion disorder called Williams syndrome. So Williams syndrome comes about due to a missing piece of DNA on chromosome 7. And you can see here, these areas at this end, these blue boxes, are areas where the sequence on that chromosome is really similar. So they're called low copy number repeats. And that sort of similarity of that genetic region means that when the chromosomes line up on meiosis to sort of share information, that sometimes they line up off kilter, which causes a portion of the chromosome to be looped out. And as a result, almost everybody with Williams syndrome has the same missing piece of DNA. They're all missing the exact same genes. And so the gene in the middle, elastin, is the one that is most closest to my heart, but it also causes a set of conditions related to the other genes within the deletion. And I like to put this slide on from a Nature Reviews that we wrote about this condition because I think it's important that this condition, although we're going to talk about it from the cardiovascular disease perspective today, touches every clinic in the hospital. There really isn't a specialist that doesn't interact with these patients. And that is important because one of the very important things about this condition is that the individuals with it, because of their cardiac disease, are at risk of sudden death with anesthesia. And so it's really important that all of the clinicians that take care of these patients know what the health concerns are. Some of the other things to sort of point out for people sort of learning about Williams from the first time is that the cardiovascular disease is important, but people also often hear about the social aspects of this condition. So it is associated with intellectual disability, but also this hypersocial personality. So most of the folks that we interact with are very friendly. They, however, have difficulty deepening social relationships. And so they're often described as having a cocktail party personality. Talk, 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 talk. But they have a hard time sort of picking up on sort of the social cues. So if I were to be talking to someone and I would say, oh, look at the time, I look at the time, but they wouldn't sort of assume that I was trying to get out of that conversation at that point. So a very interesting sort of social dynamic and sort of other health concerns that are associated with the other genes in that area. But like I said, the focus of today is cardiovascular genetics. And so cardiac genetics, in terms of the elastin insufficiency, I showed you that blood vessel before. And so when you look at blood vessels, they're made up of this little set of layers where you have the endothelial cells that line the blood vessel tube. And then you have elastin, and then you have smooth muscle elastin, smooth muscle elastin. And basically what happens with this deletion is you've removed one copy of elastin, so you have half the normal amount. And for whatever reason, it seems like the body tries to make up for the fact that it has less elastin by just making more layers. And so that layer leads to, as you can see in the bottom here, sort of a thickening of the wall where you have increased layers of smooth muscle and elastic lamellae. And so that leads to a blood vessel that is narrow, is stiff, and doesn't sort of respond appropriately. Now that narrowness and stiffness happens everywhere in the body, but there are certain areas where the blue area here is that where you get sort of focal stenosis or discrete stenosis. So we call that, when it's in the aorta, supravalvar aortic stenosis. You can see in the background of this picture the sort of wiry areas, which are stents. So this person also had a stenosis in their pulmonary arteries. So we see that there. You also see stenoses in other places, like within the vessels that go to the gut, within the renal arteries. So those stenoses can occur in places that we don't regularly see with an echo and sometimes need a little bit of sort of clinical expectation that there may be a problem there to go looking for them. Hypertension is also really common in this population. I would say the bulk of folks are at the 90th percentile if they don't quite make it to the 95th, but blood pressure is a very constant problem in this group. So when we take care of people with Williams syndrome, I originally started at WashU building a Williams syndrome center there, and that's actually what took me to the NIH, is that I developed a research program in which we studied multidisciplinary approaches to care and health of people with Williams syndrome. Now at Nationwide, we're bringing that back and growing what was a wonderful clinic based in developmental pediatrics for Williams syndrome, and expanding it to include sort of all health systems. So this is the program that we're working on there. But it's important to know that we're not the only Williams Center in the country. There are 9 other centers. When I started in St. Louis, we were actually the furthest west of any of the centers, but now we have one at Stanford and one in Texas, which is good. But it does mean that there are aspects, availability of sort of expert and multidisciplinary care for people with Williams syndrome in multiple places in the country. The other thing I wanted to point out is that the Williams Syndrome Association is very active and has a group that we've worked on, which is called CRUISE, the Collaborative Registry for Williams Syndrome. And if you have patients that you take care of that have Williams Syndrome, they have the opportunity to join this program, which allows us to collect information about people with Williams Syndrome from all over the country. Using a couple of different methods. So people who belong to one of the Williams centers can come into the Williams Center and we enter data directly through the health record for work that we do. But families who are in other places can also self-enter information. And this allows us to have a huge platform over which we can study aspects of Williams syndrome that would be hard to capture at any one particular center. It allows us to look at some of these anesthesia looks, allows us to look at sort of long-term consequences of the condition in ways that we wouldn't because people are sort of scattered across the world. So if you have patients who might be interested, please let them know that these sort of resources are available. So during my time at the NIH, we had the opportunity as part of the clinical center there to fly people in from all over the country and do broad-based natural history associated with Williams syndrome. And we were able to tackle a lot of different areas related to Williams syndrome. And I'm not going to get into all of those today, other than to say that we do see aspects of health being covered, even from a connective tissue process in multiple organ systems. I wanted to touch on a couple of things here. One, briefly, although it's not cardiac, it's cardiopulmonary, I wanted to talk about sort of the lung disease in Williams syndrome or in elastin-mediated disease, which we don't think about a lot. So I told you that elastin is 50% of the dry weight of the aorta. It makes up about 30% of the dry weight of the lung. So still a pretty significant component of the protein within the lung. When we did PFTs on people with Williams syndrome and we did CT scans on people with Williams syndrome, we found a fair amount of evidence of air trapping. So abnormal, basically that the connective tissue in the lung is allowing air in, but doesn't let it back out. And so we saw that both in the people, in particular as people got older, and then also in mice where we could look at sort of the biomechanics of that in a more specific way. We don't yet know what the long-term outcome is, although sometimes when you do see people at the family conventions, we do see older people, sometimes on oxygen and other things like that. Don't necessarily know yet about all their history, and that's part of the reason of doing these sort of larger natural history goals. But we definitely recommend that people not be in households that smoke or not smoke themselves. Because sort of the turnover of elastin related to cigarette smoke is known to be particularly problematic in this cohort. So just sort of something to put out there. And also to sort of point out that in addition to the blood vessel issues we're going to talk about, that there are issues in other connective tissue areas. So in our ligaments, in our skin, in our bladder, colon diverticuli. So we often see, unfortunately, people with Williams syndrome having diverticular disease that presents in an ER in someone in their 20s or 30s. And people are not often thinking about diverticular disease in people of that age. And unfortunately, we've lost some individuals because their disease wasn't ascertained early enough. So just sort of knowing that that's a possibility in the folks you take care of is important. But one of the main cruxes of the work that we do is related to sort of understanding the risk of sudden death in this in this cohort. So people with Williams syndrome in general have a 25 to 100-fold increased risk of sudden death at baseline. That's 300-fold, however, when associated with anesthesia. So the risks in this situation, and there are papers that have sort of tried to go through and identify what the main sort of risk factors for those are, and those would be sort of age, with young children being at higher risk, people with bilateral outflow tract obstructions. So if you have both stenosis of the aorta and the pulmonary, that is higher risk. However, unfortunately, in being the situation that I am on the research side, we often received autopsy cases from people all over the country who have had unfortunate events happen. And I won't say many of the times, but a good number of the time, the people who are having these events don't have those risk factors. So I want to caution you, if you are taking care of these patients and you're thinking about risk, that if you say, oh, they don't have SVAS, they'll be fine, and that, you know, getting those ear tubes placed, take a little bit more caution with that situation. And I always recommend, no matter what, having people touch base with their cardiologist. Always recommend having those procedures, even some of these dental procedures, being done in a hospital setting. So when we are thinking about risk factors for sudden death in this population, we're actually taking a sort of much wider swath. So some of the things that we found in looking at lots of people with Williams syndrome, like I said before, is that hypertension is prevalent. Almost everyone has a blood pressure higher than we want it to be. So we sort of draw this line is that the 95th percentile is their left ventricular hypertrophy. Yes, in terms of treatment, but almost everybody is sort of towing that line. If they're not across the line, they're right up next to the line. Almost no one, I don't remember the last time I've seen a person with Williams syndrome whose blood pressure is at the 50th percentile for age. The other thing that we see is vascular stiffness. So I told you that those blood vessels get narrow and they get this sort of thicker wall. And so that leads to differences in the rate at which blood flows from A to B. So we do a test called pulse wave velocity that looks at how fast blood moves from one spot to another. And you can think about, to a certain extent, when you kink a garden hose, how the water comes out faster and harder. It's kind of what we're seeing in this situation. And that has a particular effect when you measure it instead with this factor called augmentation. So augmentation has to do in sort of the math of how you get blood back to your coronaries. So you know that your heart, your coronaries, your heart itself is fed during diastole. So your heart pumps, blood should go out. It goes out, it hits the vascular wall, and in particular areas of branch points. And a part of that blood hits that branch point and bounces back. And it's actually that bounce back flow that feeds your coronaries during diastole. If that pattern of the stiffness and the timing of that flow is abnormal, then the blood that's coming back to feed the coronaries doesn't hit the heart at the time when it's to be delivered. We know that augmentation is significantly different. It's actually one of the most key differences that we can measure in the blood vessels of people with Williams syndrome. And so we know that that's a fact. We also know, like I said, that it's not just the SVAS. It's not just that pinch at that one very significant area, that all of the blood vessels are narrow throughout. And so you think about a situation, in particular with induction of anesthesia, where you have a person who has high blood pressure, stiff blood vessels, where the blood's coming back at the wrong time, and you acutely drop the perfusion head with anesthesia. That changes the flow that's coming back to those blood vessels at a very sort of sensitive time. And one of the other things that we found is that obviously echo, when we look at blood vessels, can look at sort of the structures very close to the heart, but it doesn't look deep down. And even when you do CT angiogram, if you're just looking at it in 2D, you're seeing there's a circle, there's a circle, there's a circle, it's open. But what you don't see is this, which is this very significant sort of tortuosity that we see in those blood vessels, where you get these hairpin turns and kinks of those blood vessels. They look open when you look at it in 2D, but again, you think about that sort of combinatorial effect of blood flow dropping that perfusion, and all of a sudden those kinks, that resistance in the flow makes a difference to what happens next. And I had to take one of the videos out due to sort of moving large amounts of data. But what we've been able to do with light sheath microscopy is be able to look down to the capillary level and see that we're actually seeing capillary dropout in that tissue. So you see sort of downstream effects of this sort of slightly low flow. It's slightly harder to get to the end of the line over time, which leads to small amounts of changes in tissue over time. And as we're seeing in our aging adults, where we're seeing more diastolic dysfunction, we're seeing more issues later on, that they have sort of cumulative effects. So when we think about anesthesia in these folks, we want to sort of think about that more globally. The other piece that I wanted to talk about was something that we think about in terms of sort of clinicians in clinic and how they think of their patients. And any of you in here in genetics or in cardiology, you've taken care of a person with Williams syndrome, and they come in and get their blood pressure, and their blood pressure is high, and then the family says, well, they're just anxious, because everybody with Williams syndrome is anxious. And I suggest in general, if you're anxious about getting your blood pressure done, you're probably anxious about a few other things in your life. So the question is, like, how are you living your life in terms of that anxiety? But in general, when we put halters on these folks and sort of followed their heart rate through the course of the day, it wasn't just in clinic. It's all day long that their heart rate runs high and high and high, and it even stays high when they go to sleep at night. That and it is sort of associated with what we're sort of thinking about is this question of sort of increased sympathetic tone. Are you constantly in sympathetic overdrive? Because that in and of itself has been associated with increased risk to sudden death. Mark Levin in my group did a study where we took halters on all of these folks and started to look at heart rate variability. So your heart should vary through the course of the day when you're up and active, when you're sitting and doing things, and just even from beat to beat. And what you should see is a good amount of variability. And when that variability gets smaller, that's where you start to see sort of that risk. And so SDNN has to do with sort of a mathematical calculation where you look at the amount of variability. It's a standard deviation of the sort of beat to beat variability. And that standard deviation gets smaller in these folks. You can divvy it out further into other types of heart rate variability. And in one case, you're looking at the right axis there of high frequency power, is this an assessment of parasympathetic tone. And you can see in the gray, which is the controls, you have a certain amount of parasympathetic tone coming up, almost nothing on the Williams side, which sort of suggests that much of this anxiety that we're looking for in terms of trying to treat the brain, trying to treat these issues related to anxiety, may actually be driven sort of more centrally than that. And many of the health features that I showed you at the beginning, anxiety, some of the executive processing, some of the belly pain, some of the sleep issues that we see in these folks may actually be driven more centrally by this sort of balance of sympathetic and parasympathetic tone. And again, this sort of comes back to that sort of sudden death risk in terms of which body parts do we need to think about as we think about taking care of these folks. So to sort of summarize this section, I want to sort of take away the sort of beginning that Williams syndrome is a microdeletion disorder. Almost everybody has the same missing piece of DNA. Much of the vascular disease comes about due to SVAS. And if you are a person taking care of a person with Williams syndrome, I want you to always have them seek guidance from their cardiologist when they think about a procedure. The guidelines that we have right now for who should get anesthesia are good. They're a place to start. They are not the full answer to the question, and we need additional work to sort of specify all of the sort of rules so that we can keep our folks as healthy as possible. In the next part of the talk, I want to talk a little bit about how we test for Williams syndrome. So we talked a little bit about genetic testing, and I like to sort of put it in people's minds who are not genetics and people, to sort of think about it from the standpoint of a shelf of encyclopedias, where a karyotype is looking at the shelf of encyclopedias and saying, is letter A there? Is letter B there? Is letter C there? It doesn't tell you much about what's in them. You can count them. That's about it. The FISH test, which is this test here, has you open up a specific book to the elastin gene and say, is the elastin there? It does not tell you anything else about the rest of the book. It doesn't tell you how much of it is missing. It just says that piece isn't there. And that's how we diagnosed Williams syndrome for a long time. You had to know you were looking for Williams syndrome, and you had to say, is it there or is it not? More recently, we introduced the chromosomal microarray, and I think of that, from that shelf of encyclopedias analogy, as taking that book off the shelf and opening up and saying, is chapter 1 there? Is chapter 2 there? Is chapter 3 there? Still doesn't tell you if there's a typo on page 437, but it gets much more sort of granular within those books to say, are we seeing what we're seeing? And so that's what you're seeing here on the right, is the microarray, is these hundreds of thousands of probes that we have running up and down the chromosome and counting how many copies of the genes are there. So here you can see a deletion where this sort of drops down below the line and says there's a missing piece of DNA here. Interestingly, the age at which we're diagnosing Williams syndrome has gotten lower as things have come along. Some of that just has to do with sort of familiarity that it exists. A lot of it, however, has to do with the introduction of this chromosomal microarray test. Back in the day, people had to know what they were aiming at. They had to know they were looking for Williams syndrome in order to ask the test. And so to that extent, our folks with significant cardiovascular disease actually got diagnosed a lot earlier because the features of the aorta were sort of pathognomonic for the condition. Whereas our folks with sort of more issues with growth or developmental delay or things like that were getting diagnosed much later. Now people can basically send one test, cover all the things, and they didn't have to necessarily know they were looking for Williams syndrome. But I will say that all SVAS is not Williams. So what we've talked about today is this big deletion, but there are also single gene changes, so changes in terms of the spelling of the elastin gene that are different in another subset. So this study we put out recently with Shane Morris from Texas, who was sort of looking at a sort of longitudinal study of what types of testing had been done in cardiovascular centers related to SVAS. And so if you took, in this case, they had 157 people who had been tested for Williams syndrome and were negative. They found that for 60% of those people, nobody went on ever to do another test. So they just sort of lived undiagnosed from that standpoint. In fact, only 18% of them had had their elastin gene sequenced. But if you sequenced it, more than 80% of those folks actually had an elastin gene change. So it was a pretty good hit if you do the test. There are a few other conditions, Noonan syndrome, dysplasia, Alagille, neurofibromatosis, that can have stenosis that looks similar enough to this to sort of get sent in that types of testing. But those things are relatively rare. But would you think of your way of sort of moving through that diagnostic odyssey for people? I do an array, it's negative. I sequence elastin, it's negative. I send a gene panel, it's negative. Maybe we'll just send the genome. So when I think about that sort of piece, and again, sort of going back to that for the folks that sort of think about these testing, single gene test, open up the book, read the whole chapter on elastin, look for typos. Panel test, or in this case, I think of sort of like the exome, which is the all of the coding genes. I think of that if you took the Cliffs notes or the Spark notes to the encyclopedia and read every letter of that book looking for typos, that would be that kind of test. Whereas the genome is taking every single one of those books of the encyclopedia off and reading every single letter in it and looking for typos. There's a lot of information in that. But I think that's where we're sort of going in terms of being a general practice for what we're going to start to take on. And right now, I think in general is the time to be able to do that. The price per genome is going down. The analysis time is also going down, while the number of conditions that we can diagnose, as Vidu just showed us in terms of bicuspid aortic valve, all of these genes that we're starting to accumulate is going up. Most of the time, or a good percentage of the time, we're actually able to do something in terms of medical management or altered outcomes for our patients. And so I think in general, it is the time to start considering that as our sort of first line test. We began offering genome sequencing in terms of in-house testing this year at NCH, and we were able to sort of deliver that test on a rapid basis back to patients within 3.5 days of their having their test sent out, such that we can take a patient in particular in our ICUs, figure out what's going on, and sort of deliver that information back to them. Right now, our diagnosis rates in this case are 25%. That number is going to go up once we have copy number affiliated with that, which right now it isn't with our test. We do that separately. And we're going to see more and more of this as time goes on. And right now, we're working on an implementation strategy to sort of bring that to our whole hospital in terms of being able to do all of these rapid diagnostics in our ICU, to partner with all of our outpatient clinics and cardiogenetics to be able to get the testing that we want. And to move towards therapeutics. And so what I want to focus on in the last few minutes is, as much as I can get through, of a couple of sort of research aspects of thinking about, well, if we can do the genome now clinically, there's a certain part of find the gene, find the answer, but what else can I find from that whole swath of data I've collected beyond the one base pair that sort of makes the diagnosis? What else can I do with that information? So we're going to try to talk a little bit about modifiers, trying to unravel variants of unknown significance. And if we have time, we'll get into a couple of the other areas. So like I said, I take care of lots of people with Williams syndrome. And the thing that has always struck me in taking care of people is that no two people, even with a rare condition, are the same. And when we started doing that deep phenotyping and putting all of those things together, we found that there were sort of categories of health that people sort of fell together in terms of the health risks that they had. And so we became very interested in trying to tackle that sort of question between, single gene, single answer to your disease, and that sort of combinatorial effect of genes. So even in SVAS, which is sort of like the board question answer for Williams syndrome and elastin-mediated disease, not everybody has it. They all have the same missing piece of DNA. Around 20% of them have SVAS of significant severity that they're going to need a surgery for it in early life. But there's a full sort of 40-ish% of them that really don't have much SVAS at all. And then there's a group in the middle that has sort of mild to moderate disease. So all with that same, I'm going to call the elastin gene in this case risk factor, but not everything that you need in terms of getting to an SVAS phenotype. So when we think about other things that might be responsible for that, I'm going to skip over a bunch of it, but I can tell you we've looked across all of these things. So were there slight differences in the deletion size that had an effect? Yes, for hypertension, no for the presence of SVAS. Are there variants on the remaining elastin allele that make a difference? I'll tell you that there's almost none left. If you delete one variant of elastin, that other elastin allele tends to be pretty clean, which we'll talk about a little bit later. Second, sort of rare diseases, yes, but rare. I have a couple of patients that actually have Williams syndrome and Marfan syndrome. That certainly will affect your aorta in different ways. And certainly other rare conditions can happen cumulatively. Changes, and so then where we sort of focused our efforts are on risk factors that sort of cumulatively come together to add up to having a disease consequence. So the way that this has been done very simplistically is with association. And this is a very simplistic way to show association. You take a single variant and you look and say, does this happen more often in the people that have the bad thing from the people that don't? And you look for sort of a mathematical association between those things. And most of the time when these things like GWAS are done, they are done in tens, if not hundreds of thousands of people. But I work in rare disease, and there will never, ever be tens to hundreds of thousands of people for whom I have genomes on. And so we needed to find a more creative way to sort of work through this process. And the way we did that was working to develop a method where we would take all of the variants from a group of patients who had severe or surgical SVAS and the people who had no SVAS, put all of their variants together and sort them in a couple of different ways. First, in this case, we use CAD score. Now there's another score called REVEL, but basically what it is it's a score that tries to suggest, is this variant going to do something bad? Where a bigger number means more potential for badness. And so we separated them that way on the x-axis. On the y-axis, we separated them based on the allele frequency, the frequency of that variant in people who had the bad disease versus the people who had sort of no SVAS. And we selected out just the purple ones. So those are the ones that have some degree of pathogenicity and are uncommon in one group versus the other. And we decided that we thought that it wasn't going to be one thing, but in fact, a cumulative effect of hitting the same pathway over and over again. And so we did a thing called gene set enrichment, where we took all of those variants and said, what do they have in common? What sorts of things are we hitting pathway again, again, and again? And when we did that, we found 4 things that we hit over and over again, and they made a lot of sense. So one was you hit the extracellular matrix. Elastin is a matrix gene. If you muck with the extracellular matrix in other ways, that causes problems. The second thing is in immune. We also saw lipoprotein and some G protein signaling. And interestingly, there had been other papers already put out in terms of people doing sort of combinatorial gene effects that showed that these pathways made sense. This, however, was done in a relatively small group of people, so we went back and replicated it in 5 times more people and found the same answer to the question. So hitting these same pathways, hitting the matrix, hitting the immune system a second time, in addition to having the elastin gene deficiency in terms of the deletion, led to more severe outcomes. We also found when we looked at those matrix genes that they weren't just any matrix genes, but it turns out that the ones that come up through this analysis are things that are actually active and important in blood vessels and in the aorta. But we wanted to be able to do something with this. And so we thought, well, can we take these genes and put them together in this sort of polygenic risk score, something that will predict who's going to have a severe outcome? And so basically what we did is we took 2/3 of the data and lumped it together and said, which ones of these variants are most important? And then tested it on the other third. And ultimately what we found is from that whole chunk of variants, we could come up with 322 from variants from those key pathways that almost perfectly separated the people with surgical SVAS from those who had no SVAS. We then added an additional cohort of people that were that sort of mild to moderate group. So this was a group of people whose genomes had never been involved in that initial process and found that they landed right in the middle in terms of those scores. And when you actually tried to compare what's the misclassification rate between the surgical group and the sort of mild to moderate group, there was about 10% that we missed in one direction or another, not having incorporated those folks into the pathway. Bit more misclassification on the back end, which is not surprising, as there's not as much sort of imaging that goes on in the folks that are at that sort of milder end to see if they sort of stray from being none to a little. We can do this again by taking sort of the whole set of variants, those whole purple variants, as opposed to the ones that were just within the pathways that we found. And with 622 variants, we can actually get up to 100% of perfect will separate between the no SVAS and the surgical. And can bring that misclassification rate down to 7.5%. So what I think that this suggests is that most of the conditions we take care of are multigenic to some degree. You have one big hit that sort of puts you at risk, and then multiple hits often within particular pathways that sort of up the ante and sort of push you off the ledge in terms of having that phenotype. Potentially finding what those other genes are has the potential to be able to tell us very early on, this person's at risk, which means that if we then start having therapeutics that we can put in place, potentially we can put them in place before it's dire that we need to do surgery. It also allows us to give a little bit of more information to families at the time of diagnosis beyond this is the condition, but what's going to happen. All right, so we're going to talk one more piece here about how we think about unraveling variants of unknown significance. So this diagram, which is very busy, outlines, at least as of 2017, the variants that had been described that were associated with basically misspellings within the elastin gene that cause isolated SVAS. If you look at them carefully, the bulk of them are either stop gains, so they stop the molecule, or frame shifts. And lead to, therefore, haploinsufficiency for elastin, so half the normal amount. There are really not within this scope of variants missense changes, so things that are a misspelling in the gene. So instead of having less elastin, the expectation might be, what happens if you have bad elastin? You have poorly spelled elastin. And so we wanted to sort of solve that question, as well as asking about, in particular, questions related to splice variants. So elastin is an interesting gene in that all of the exons are actually in frame with each other. And so if you skip an exon, it isn't necessarily bad. And so what happens at a splice variant was something we were also interested in. As I told you at the beginning, when we look at people with Williams syndrome and look at sort of the common variation that exists on our genomes, we found that people with Williams syndrome have almost none, which suggests when you only have one copy of the gene, your other one better be pristine. So there are really only two variants that exist in the background of people with Williams syndrome, two common variants, one that's present in 40% of the population and one that's present in 10. And those are expected to be benign. They exist in the world, but that's pretty much all the other variants that you see on that background. So we wanted to look at what these other sets of variants might do. And to do that, we wrote a UO1 grant with Mark Williams and Nephi Walton when he was at Geisinger to look at what happens if you take a genes first approach. So normally when we do this, we as geneticists look at a phenotype and say, could this be genetic? Let me send the test and find out. This sort of takes the opposite approach. It's let's look at a bunch of people, figure out who has the gene, and see what phenotypes they have in common. And so that was the approach we took with this grant. In terms of the variants we selected, we wanted sort of rarer variants. So in this case, they were variants that were present in less than 1% of the general population. We had a preference for missense alleles and things that were adding charge to the hydrophobic domain, so things that were sort of important to the molecule, and variants that would impact splicing. And so this is basically what we had. They, at that time, so their hospital consents everyone that comes into the hospital for sequencing. You can opt out. And we, at that time, they had sequenced, in this case, exomes of 184,293 people. And we said, give us all of the people in your system that have variants within elastin that are of the character we described. And they found 254 of them. 29% of those had a very specific variant, proline 220 leucine. 45% of them were other types of missense alleles, and then 25% of them affected splices. We then did because all of their genetic information is then linked to the medical record. So the team there could go back and look at all these people and say, well, what do they have going on? What's in their health? And ultimately, we found that about half of the people, we had a very sort of specific rubric of things that we were looking for based on a hypothesis of what elastin would do. And we looked at those features and found that around half of them had features within the area that we were interested. And of those, the overwhelming majority of them were in cardiovascular disease. So as we might have expected, we saw some narrow blood vessels, but we interestingly also saw a lot of big blood vessels. So we saw dilation, we saw both stenosis and dilation. We also saw other aspects of connective tissue disease. We also then performed a FEWAS, where basically we took all of those patients and compared them to a whole bunch of people within their medical record system who don't have elastin variants, and just said, what do these people have in common that the people who don't have an elastin variant found? And what we found was aortic dissection. So again, when we think of haploinsufficiency for elastin, we think stenosis. But what we found with all of this missense variants was actually big blood vessels, dissection, aneurysm, the opposite effect than what we would have thought. So bad elastin versus less elastin are two different things. However, in this situation, you're using entirely EMR, so a medical records. And the only thing that's in a medical record are the things that somebody went to the doctor for. So while there is a benefit of having this huge data set, a huge amount of phenotypic data, and a broad window in which you can look at people, you don't really have a denominator. So I, happily, have never had a reason to have an echocardiogram. I have no idea what my aortic size is. And an EMR that was looking at me wouldn't know the answer to that question, even if I had an elastin variant. It also doesn't track information that has happened outside their particular health system. So in this way, it is impossible to distinguish, we've never looked, from within normal limits. And we wanted to know the answer to that question. And so for the aspect of that, we were able to call up these folks and say, would you like to come to the NIH and find out? So we worked with genetic counselors who called up the families and said, you have a variant, which you had consented for. There is a research study if you would like to be involved in it. And you can come down and shuffle people's benefits. So far, we've brought 20 people to the NIH. One of them is actually there this week, so I don't have their data. But we basically were able to do a full set of sort of phenotyping on them during a week at the NIH. And some of the key points to sort of start was that the bulk of the people who came to our study considered themselves healthy to begin with. They were just people in the world that were just sort of interested in a trip to DC and sort of seeing what was involved, since they got sort of this call that something might be different with their genome. None of them had in their medical record a diagnosis of SVAS or Q dysplasia. But ultimately, 100% of them went on to have variation in terms of their phenotype that may be associated with elastin gene changes. So when we look, in this case, I have 18 of the variants in here as we get all of our data together. But when we look at the people with splice variants, their data looked like sort of a hypoform of SVAS. So it looked, the blood vessels were small. And we did have one person that was big, but the bulk of them had small blood vessels. We had one person with a variant in the UTR, but all of those, I think, are sort of acting to some degree like gene dosage. They're affecting the splice, they're affecting the total amount of protein, and are leading to a sort of small vessel phenotype. The people with missense variants, however, are the people that we were finding that are having aneurysms. And ultimately, we found several entirely not in people's medical record aneurysms when we went and sort of scanned these folks a priori. It's exciting. But we were able to get that information back to the families and get them hooked up with people to be able to sort of manage and follow these things long-term for them. But we found it in a reasonable number of people that we saw. We also saw other more common things, so tortuosity to the vasculature, some anatomical variants. Yeah. Anatomical variants are variants, but it also suggests that genetic and genes also affects traits. So it doesn't necessarily mean that there was a physical impact, but it means that these genes are responsible for the way that our bodies form. We also see a lot of hypertension in this cohort, and hypertension is common in the general population, but the amount of hypertension that we saw in this cohort was higher than you would expect based on the age and sort of demographics of these folks. We saw more rhythm disturbance than we might have otherwise expected, which we've also seen in our Williams cohort, in particular in the aged group. Some family history of sudden death. We see some skin lesions in terms of stretch marks and altered skin elasticity. And interestingly, a fair number of spinal fusions, which are relatively uncommon at baseline, but we've seen it in a fair number of the folks that we've seen so far. We are working with a group at the University of Connecticut to do modeling in these, where we can sort of look at what happens in particular with these missense changes. How does it change the molecule? And ultimately, this particular variant, it may be sort of difficult to see what the radius of gyration means, but it ultimately means it leads to a sort of a molecule that holds together less stiffly, so it's sort of more widely spread. And all we can sort of guess or ascertain at this point is that that leads to an elastin molecule that's a little bit sort of more loose. But those are things that we're still continuing to work on in the lab. But to me, this sort of leads to this thought that there is a gradation in variant effect from these sort of high impact variants that take you most of the way there to these sort of moderate effect variants that may act more as sort of like risk factors for things as we age over time. And then the sort of more common benign variants, and that we need to think about these things as we start to look at genomes. How are we going to deliver this information? What are we going to do with those risk factors? In this case, scanning some of these folks that we did at the NIH because of their variant may have led to pretty significant changes in their lives. So do we report those things or do we not? And that's going to be something that's going to be changing over time. So in conclusion for this, I think large data sets are available, and you may hear about more of those later today. Reverse genetics will tell us a lot of things about what these variants might mean, but we're going to need a lot more data in terms of in silico modeling and other things to fully understand this impact. In the last couple of minutes, I'm going to try to cover one last question, which is sort of solving missing heritability using a genome and additional technology. So in this case is one of the folks, I showed you that pie graph before, where we have a group of Williams, we have SVAS, we have a few other syndromes, and then we have this group of people that are sort of stubbornly undiagnosed. And so the question is, what are the genes, what are the features that are associated with that form of SVAS? So this, unfortunately, is a case of a little one who was diagnosed with a murmur, went in for evaluation, and had a sedated echo at 9 months of age, and unfortunately decompensated during the recovery associated with that. He had SVAS with a thickened ascending aorta with a Z-score of -4.8, a stenotic main coronary artery, as well as long segment narrowing of the descending aorta, a diffuse PA, and branch pulmonary narrowing, as well as LVH. He had elastin gene sequencing with no variant identified and had 2 copies of the elastin gene. And so in terms of, obviously, this terrible event, families wanting answers, wanting to understand recurrence risk, wanting to understand sort of what's going on here. Interestingly, though, what we found was although they did not have a deletion that included elastin, they had a deletion next door. And so I showed you before in the Williams deletion, elastin's right in the middle. In this case, they had a deletion that was sort of the left side or the centromeric side of that, and took out some of the genes nearby. We actually, interestingly, also got referred to another patient who had that, a similar type of deletion. And then through going through ClinVar, we're actually able to find 4 cases altogether of people who had these deletions that didn't cover elastin, but did have SVAS. The ones on the bottom are a group of patients who have deletions that are sort of further to the left or further centromerically that didn't have SVAS. And then there were a group of 3 patients within ClinVar that had deletions that covered our area of interest, but there wasn't enough information to say one way or the other from those entries what was going on with those patients. So from the folks that we actually met, we actually had connective tissue, or we had tissue from those patients. And we were able to do a test that relies on that common variant that I told you about. So I told you there's these 2 common variants that are present in the general population. We don't think that they do anything bad, but it allows us to be able to do transcriptomics. So take that tissue, in this case skin, and look and see if you're making elastin off of both alleles. So we can say, oh, the one with the T is making one, the one with A is making them. And so in this case, when you look at control fibroblasts, you see that they're making elastin about equivalently off of both alleles. When you look at people with Williams syndrome, they only have one, so they only make one. But people with SVAS who have stop gains or things like that within the gene are only transcribing off of both alleles. So even though both genes are there, you're only seeing transcript in terms of mRNA from one of them. Our other patients that have these atypical deletions have a fully intact elastin gene, but are only transcribing off of a single allele. So that suggested we don't need to look for the whole rest of the genome potentially to find the answer to the question. We need to look more carefully sort of close to this gene. When we looked at that allele, we also used long read sequencing to sort of collect the deletion to which variant it was, to say that it was the one that is upstream of the deletion that's not being made. Oops, sorry. I'm trying to stop myself. And so that sort of solidified that piece. And so that made us think about enhancers in terms of what might be happening in that upstream region. And ultimately, if you use some of the available databases, we were able to find a couple of areas within that area of interest that seemed to be enhancers, in the sense that they were parts of the gene that were important, or parts of the genomic region that were important, and sort of available in tissues that make elastin, but not in ones that don't. And so what we ultimately did, we serendipitously also found a patient that had a very small deletion that covered that area, which had been in a group of patients that had been seen clinically but not reported out because nobody knew what to do with that piece. And so we made a mouse related to that. And I'm happy to tell you that the mouse genes are sort of lined up the same way that the human genes are. And when we delete this upstream region within it, we basically get that SVAS phenotype back. When you look at the tissues, you get increased lamellar number, you get a longer aorta, a narrow aorta, stiffer aorta. And so again, this would have been FISH negative. This was a CMA that people didn't really know what to do with because it didn't cover the elastin gene, and it wasn't an elastin gene change. But it is potentially information that we could get from a genome if we sort of understood where and what to look for. So again, there's missing heritability for elastin-associated disease, and it requires sort of careful curation of patient data. Enhancers are understudied and are likely to contribute a significant portion of unsolved disease. That information all lives in these genomes that we're doing, but we're going to need the technologies and the ability to sort of look at that in order to pin it to be the answer. So again, this comes back to optimizing implementation for how we do genetic testing. We're working at that on a clinical basis, trying to put genomes into the care plan for as many of the patients as we see. But obviously, there's still a lot of room for research in terms of sort of bumping that line in terms of what is sort of clinically known and clinically accessible. To conclude, I want to say, obviously, we did a fair number of things, and it involved a lot of different people that needed to come together from a lot of different places. And ultimately, that is how we're going to do genetic and genomic work going forward, is only through the sort of collaboration of large groups are we able to get to the answers that we want for our patients. So that concludes my talk. I'm happy to take any questions. Thank you. Julie. So I will say we all of our folks actually see a person in sort of a musculoskeletal and does that sort of full evaluation, and they do not widen out to look like hypermobile ES. That said, we have had people, most of our patients are referred from Geisinger and come straight down, but we have an arm of the study where people can self-identify. And so we have had people who have had elastin variants that were identified on a panel for sort of more connective tissue-y things that are coming to see us. So we're trying to assess that. I would say within that category, I don't think that this gene, even though it seems like it could be, is sort of a sure thing, at least in terms of what we're seeing for those variants related to hypermobility. But I do think we're going to need a lot more data over probably a lot more of these databases to say for sure what is the sort of implication for going and starting to do testing for all of that type of cancer. So I would say it makes me nervous, and I want to know, but I don't know that I have the data to say open the floodgates for that. Yes. Yes. So some of the, just to sort of recap the question related to some of the things that we brought up in terms of sudden death, but is that related in addition to things that we might see for myocardial infarction or stroke or things like that. I would say we're not seeing those things in the pediatric population outside of events that are associated with surgery and pump and things related to that. In the adults, and this is part of the reason that we sort of set up this registry, is that many of our adults get lost to care from primary centers after they leave pediatrics. And so I would say that our data for what happens later is poor. That said, when we look, one of the other genes that's associated with Williams syndrome affects the amount of sort of lipid versus blood sugar issues that you have. And we actually, even though the bulk of the folks I take care of eat a diet of pizza and chicken nuggets, they honestly have relatively okay lipids. So we aren't seeing a lot of those things sort of like on top of the stenosis parts of things. But what we are seeing as we've sort of followed these folks out for sort of longer effects is less, straight MI and more gradual decrease in function over time. And like I said, in terms of strokes, I think they probably are higher for age than the general population, but the numbers are still really small. And so again, that's part of the reason that we would love to be able to get as many people into these registries that we could be able to capture that data better. If you yell, I'll repeat it.
It's a great talk. So I had a question about the breath.
So in the patients that, the one infant passed away, so we didn't have a lot more to go on. And the older patient that we saw, their phenotype was atypical for Williams syndrome in the sense that they were missing the whole right half of their deletions. So there were parts of the Williams syndrome that they did not represent. When we looked for that sort of atypical or asymmetric splicing, or not splicing, but expression of other genes further on, we didn't see any difference in those genes, suggesting that this enhancer was at least mostly specific to elastin. Whether things could get further away, I don't know. But I would say from the standpoint of the vascular disease in particular, it looked pretty typical for elastin-mediated disease.
Thank you for the wonderful talk.
Oh, it's a good question. So the question is, in particular, young people with hypertension and nothing else that you can sort of pin it to. Should we start be looking for genetic things? My bias would be yes. But it's interesting from the standpoint of, so the Baby-Seq study that came out where they actually started sequencing kids in sort of preparation for, can we use genome for the newborn screen kind of a thing. Actually, elastin came out as one of the top variants that came out of this. And so from that standpoint, knowing and being able to sort of screen for additional risk might be beneficial. And in that case, knowing sort of what else you're up against. And I would say from the standpoint of, certainly I would recommend in the hypertension case, I would hope that in that situation, people are imaging, but you're also at least getting your renal ultrasounds and you're getting other things that would have sort of suggested other sort of comorbidities. But I would say certainly in a situation where you're seeing anatomical changes that might suggest and elastinopathy, I would love to see you testing for, because there are certainly patients that we see who have just renal artery stenosis and don't have the other pieces of it. And it's not uncommon. Actually, there's a study that was done looking at where they tried to suggest that people who have isolated SVIS have worse vascular disease than people with Williams. But I think it turns out to be sort of an ascertainment sort of thing, because people are ascertained for Williams for intellectual disability and other things like that, whereas you don't get sequenced for elastin if you don't have the vascular disease. But what they found when they went back and looked at those people's parents is that it was often that the parents had a minor something. And often hypertension was one of those things. The kid came in with a very bad vascular disease, but the parents actually had sort of signals of having had a genetic condition prior to that. So I might say start with your first round of doing some imaging and making sure there's something there. And then I would sort of be biased toward looking for both the connective tissue reasons and other reasons of sort of vascular. Thank you.
Great. Thank you so much to our speakers this morning. It has been wonderful. We now have time for about a 10-minute break. It's 10:04. Please get up, use your legs, use the bathroom, visit our exhibitors. Thank you. Perfect. Dr. Ware is the chair of the Department of Medical and Molecular Genetics at Indiana University. She is a highly respected physician scientist in the field of genetics, and she is currently a professor of pediatrics and medical and molecular genetics at IU School of Medicine. She serves as the vice chair of clinical affairs within the Department of Medical and Molecular Genetics and is the director of cardiovascular genetics in the Herman B. Wells Center for Pediatric Research. She is a graduate of Butler University with a Bachelor of Science in Zoology. She earned her PhD from the University of Cincinnati College of Medicine, followed by her MD. She completed a residency in pediatrics at Baylor College of Medicine and a fellowship in medical genetics from Baylor. Dr. Ware will be speaking us today on the topic of heterotaxy.
Well, thank you, and it's really nice to be here. I was asked to talk about heterotaxy, and that's something my lab has worked on for a long time. But obviously, this is a more clinical audience, so I'm not going to talk really about any of our basic research work, but wanted to bring up the topic of genetic testing and heterotaxy, and then just some of the actual problems with the diagnostic criteria for heterotaxy that make it difficult to do good studies related to it. So as many of you probably know, heterotaxy is a disorder of laterality. And so shown here is the normal anatomy, also termed situs solidus. And when you have a complete mirror image reversal, the terminology for that is situs inversus totalis. And then there are multiple different arrangements that you can have in terms of your organ positioning that are neither situs solidus or situs inversus totalis. And in my mind, heterotaxy applies to all of those in-between conditions, but there are a lot of different terms that have been used, including situs ambiguous, left isomerism, polysplenia, right isomerism, or asplenia, Ivemark syndrome, situs inversus abdominalis, situs inversus thoracalis. And so this number of different terminology has actually made it a little complicated to talk about heterotaxy. And even though the initial thought was that it would be helpful to classify them, help people think about it, but I think it actually confuses the picture. And the more important thing clinically is really to understand the anatomy. So if we look at the epidemiology of laterality defects, this is probably the best study out there on it. Angela Lin, many of you may know, is very accomplished geneticist and cardiologist at Boston. And she led this work by the National Birth Defect Prevention Study, where they looked at the birth prevalence and descriptive epidemiology. And this paper came out in 2014, and I quickly dug into it. And I'd been studying laterality disorders for about a decade at that point. And the first thing I noted was that when you look at congenital heart defects in heterotaxy and then in situs inversus totalis, they look at both simple and complex. And I was very jarred to see this because in my mind for a decade, I had thought of situs inversus totalis as being a complete mirror image arrangement of the organs, which meant that you would have dextrocardia, but no structural cardiac abnormalities. And so I was immediately confused by the fact in this large epidemiologic study, situs inversus totalis was showing that 14% of patients had a simple cardiac defect defined as either an ASD, VSD, mild pulmonary valve stenosis, or mild aortic stenosis, and 26.6% had a complex heart defect. So I thought, well, how can I have been studying this for 10 years and I don't seem to know the definition of situs inversus totalis versus heterotaxy? You'll see here with heterotaxy, then the number of complex cardiac defects is much higher, 82.8%. But there are patients with heterotaxy that have no congenital heart defects. So this was, in this study, the way that they defined laterality defects, the inclusion criteria, and it's kind of complicated to read on a slide like this, but basically they laid out a number of characteristic congenital heart defects, a pretty comprehensive list there on the far column. And then in addition to that, related to the heart and vessels listed separately are the morphology of the atrial appendages, which is number 7, and then the systemic venous anomalies listed as number 8, which would include bilateral SVC or interrupted inferior vena cava. So in addition to that, the other features that you pay attention to is whether there's biliary atresia, the abdominal situs positioning of the liver, the stomach, the spleen, any spleen abnormalities in terms of asplenia, polysplenia, and then lung isomerisms, particularly of the bronchi and the lobation of the lungs. And then finally, intestinal malrotation and gallbladder presence or absence. So in this study, the definition of heterotaxy required at least 3 features from group 1 to 8, which is a pretty stringent definition, but very well done in this study. So, last, I guess the end of 2022, a group of us published this multidisciplinary approach to management of children with heterotaxy. And I think it was sort of past due in the field because there are so many specialists involved in taking care of these individuals and to kind of put out some standard of care guidelines. But in reviewing the epidemiology of laterality disorders, it brought to my mind, well, can you provide standard of care, best practice management, if you don't have a general consensus on what is heterotaxy, what the definition is? So I provided some examples here. I think, cardiology, genetics, most of the subspecialty groups that care for these patients would agree that a patient with an unbalanced AV canal, interrupted IVC, arterial bronchi, asplenia, a right-sided stomach with microgastria and midline liver is sort of a classic heterotaxy patient. But, what about a patient with double outlet right ventricle and mitral atresia, LTGA, and a malrotation, but normal situs of liver, spleen, and stomach? For me, from a research basis, this is clear evidence of a laterality disorder. According to the National Birth Defect Prevention Study, this does not meet criteria for heterotaxy. So what do you call this patient? And then another example, a more simple heart defect, atrial septal defect, but left-sided liver. Again, clear evidence of a laterality defect and maybe not being given a diagnosis of heterotaxy. So depending on who sees this child, are they going to be recommended for more diagnostic workup and imaging? Are they going to be recommended to have functional studies for spleen status related to risk for infection? And then, the last example is just malrotation and polysplenia. So, two organ systems involved, again, evidence of laterality disorders, but the heart not involved here. From a research standpoint, we would clearly call this heterotaxy. From a clinical standpoint, I think the subspecialist involved in caring for that patient, probably that patient would never get a diagnosis of heterotaxy. So, what are we going to include in this? And then does it really matter? Well, I would argue that it does matter for a couple of reasons. First, we know that the coding for heterotaxy is not precise, and it's very, very difficult to standardize across centers with regard to registries, any kind of natural history study, and that makes trying to understand outcomes almost impossible. This is a rare disease, and even if you have multi-center studies, you're going to have a relatively small number of patients, and they're all going to have different anatomy. And so it becomes very difficult to ask and answer good questions with regard to clinical care and outcomes. And then, I think it's not secondary to think about the patient or family experience in this regard. So there are so many times that I've heard families talk about diagnostic surprises. The 20-year-old who has had a lifelong diagnosis of primary ciliary dyskinesia that has just learned that they had visceral situs anomalies. So really they have heterotaxy. Individuals with heterotaxy who have had infections their whole life and they were never noted to have spleen abnormalities because that part of the workup just wasn't done. The other piece of the patient family experience that I hear a lot is a sense of isolation. And I think that often they may hear a specialist say, well, I've never seen this anatomy before. And that's not meant to be isolating. It's meant that, that there are complex malformations and complex combinations of malformations in patients with heterotaxy, but often that's heard by families as, I've never seen anything like this. I don't know how to take care of this type of thing. And then finally, I think without having a common definition of heterotaxy, it requires the patients and families really to become medically sophisticated and be the ones that advocate for seeing subspecialists that patients with heterotaxy should see, but if they haven't really been given that diagnosis, may not be occurring. So that's an argument for some type of consensus around the naming of heterotaxy, the diagnosis of heterotaxy, and potentially the establishment of national registry that would help us better understand the spectrum of this disorder. So if we then look at the genetics of heterotaxy, we know that congenital heart disease has a strong genetic component. And this is from another epidemiologic study, but it's from the National Danish Registry. And we see that heterotaxy of all of the classes of heart defects has the highest relative risk. ABSD, left-sided obstructed defects and right-sided obstructed defects also have relatively high recurrence risk. So we know that it's important to study the genetics of heterotaxy, and it's fundamentally a very interesting developmental biology question because the development of left and right is something that occurs early during embryonic development. So there's actually been really good animal work done that has outlined many of the pathways that are important for left-right. And that's important for us in genetics because it gives us a number of pathways to interrogate. It gives obvious candidate genes. And so what's shown here in the first panel is just a scanning electron micrograph of the left-right organizer or node of a developing embryo. In the middle panel, you see the central node cells kind of highlighted in red, and then what's called the crown cells that surrounds it in green. And we know that there is a morphogen gradient that's set up during development. This is before organs are formed at the end of gastrulation that really establishes the body axis so that the left side is aware that it's left, the right side is aware that it's right, and then the future organs orient themselves with regard to that left-right axis. Part of the reason that complex cardiovascular malformations are so common in laterality disorders is that the heart seems to be particularly susceptible to these positional cues. And given that the heart starts as a midline tube and then undergoes a number of complicated curvatures and looping in order to form the final four-chambered structure, it's not surprising that if there's ambiguity around left and right, that looping would not occur properly. So in terms of mechanisms of heterotaxy disorders, there's at least three broad classes. The first is cilia abnormalities, because there are cilia that are present at this left-right organizer. The second is cell polarity defects, because cell polarity is important for the positioning of the cilia on the left-right organizer. And then the third is TGF-beta signaling, which is the nodal signal transduction pathway, which is important for the morphogen gradient and the establishment of the left-sided identity. So as I said, there's been a number of work done using model organisms that have really shown that these pathways are very largely conserved across organisms and are essential in dictating proper organ formation and positioning. So in terms of the genetics of heterotaxy, then this slide is a little bit dated, but there are different panels that can be sent if you wanted to send panel testing. So there is heterotaxy panel out there with a number of genes, and then primary ciliary dyskinesia, which I'll talk about a little bit more. There are specific panels for that that include over 70 genes, and then ciliopathy panels. And in terms of the inheritance patterns, from a Mendelian inheritance standpoint, essentially every type of inheritance pattern is possible. SIC3 is a known cause of X-linked heterotaxy. There are rare cases of autosomal dominant and then autosomal recessive, especially in cases of primary ciliary dyskinesia. Far and away, the most common thing that we see, though, is a complex inheritance pattern or multifactorial. So I'm going to do a little sidebar here of a case which came up recently, which I think is kind of interesting for a number of reasons. And it was one of these cases that you sort of hear about by happenstance and then are very glad that you did. So this was, I got an email about a patient that was being seen prenatally, situs inversus totalis on the fetal sonogram. And so carrier screening was done in the parents, and suspicious variants in DNA H11, which is a gene that is important for components that are part of the cilia architecture, were identified. And so this is the way that it kind of occurred, and this is a slide from my student who then presented this. So the genetic counseling team, they were interested in knowing, is it fine for this patient to deliver at the outside hospital, or is this something they would need to come in for? So they sent that to me, and I was kind of concerned about heterotaxy. How good was the imaging? Did they do a fetal echo? And, you know, we need to think about PCD. And so I said, well, let us look at the variants in more detail. And also we should involve Ben Gaston, who is a pediatric pulmonologist at our institution who runs the PCD Center. So my MD PhD student was just finishing up his graduate work and had been working on RNA splicing. So I sent him the two variants and said, hey, look at these variants and see if you think splicing is possibly at risk here. So the prenatal diagnostics where the father had a known previously described pathogenic variant in DNA H11. The mother had two variants that were actually listed as likely benign by the commercial lab that did the testing. We're upgrading one of these variants to the US now. And so these were the two that I sent to my student. And I mostly was interested in him looking at the variant that's the plus 15 C to T to see whether that might affect splicing or not. But being the good student that he was, he looked at both of them, which was great because I was very surprised. So the variant that was the plus 15 C to T was not predicted to alter splicing, but actually the missense variant was predicted to alter splicing. by causing exon skipping, which would then lead to an early truncation, potentially nonsense mediated decay, or some other type of instability related to this protein. So, based on this, we said, well, we're suspicious given we have a pathogenic variant and what we would consider a variant of uncertain significance, and a fetus with situs inversus totalis. We're suspicious that that these may be causative, and so that information was relayed to the family, and they decided to come and deliver in Indianapolis because of that. So born at term, APGARs of 8 and 9, but then within the first 8 hours of life, grunting with retractions was transferred to the NICU. An echo was actually normal, so this was true situs inversus totalis, and had a chest X-ray consistent with transient tachypnea of the newborn. A nasal brush biopsy was done for high-speed video microscopy, and I'll show those results here in a second. And so clinically, given the clinical features and what we knew about the variants, which I'll show here, the testing was then done on the newborn and showed that had inherited the paternal pathogenic variant and the maternal variant that was potentially at risk for a splicing error. We gave the tentative diagnosis of primary ciliary dyskinesia. And so that did have management implications, which may have been missed otherwise. The family learned how to do chest physiotherapy. The child went home with that. These patients are treated in a similar fashion to the patients with cystic fibrosis, where they get chest physiotherapy, they get routine sputum cultures, they get pretty aggressive antibiotic prophylaxis at various intervals. So this is from the cultured cells from the nasal brush biopsy. At the top, you can just see that there was DNH11 staining, so there wasn't complete loss of the protein. And then the high-speed video microscopy, if I can get it to work. I may need someone up there. They said, oh, there we go, okay. So this is a control, and you can see the cilia kind of waving and beating there. And then in the patient, there's pretty decreased beat amplitude and also overall fewer cells. So this provides functional evidence to establish the diagnosis of primary ciliary dyskinesia. So I think the importance of this case, and relative to the title of Do Genetics Matter, the prenatal testing or the prenatal findings combined with the carrier testing in the family allowed for positive anticipatory impact on clinical care for the patient. It also highlights the issues that non-coding mechanisms of disease, such as abnormal RNA splicing, post-transcriptional modifications, disrupting gene regulatory networks, are all the next frontier in terms of what we need to be understanding related to these kind of cases. And for my student, the good news for him was that there's a very active PCD Foundation group, and they very much want to encourage young investigators. And so he was given an award to go present this case in Puerto Rico in February, which was lovely. So, so conditions to think about in newborns with heterotaxy. There's at least 4 categories of disorders. So the first would be ciliopathies, group of disorders of primary or sensory cilia, and I'll talk a little bit more about them, as well as this category of primary ciliary dyskinesia. We don't have great numbers. I'll show in a couple slides from now work done to show the prevalence of heterotaxia-related laterality defects, but we do know from sort of family surveys, which of course have their own ascertainment bias, that at least 30% of patients with heterotaxy have occurrence of chronic otosinopulmonary symptoms. So it's definitely an area that needs more study. There is syndromic heterotaxy, by which I mean genetic syndromes that are non-randomly associated with heterotaxy. And those would include things like trisomy 13, 22q11.2 deletion, a number of other conditions. And then finally, there's isolated heterotaxy, which is a genetic condition, but for which we don't routinely find the cause outside of X-linked heterotaxy. So to say a little bit more about cilia, there are multiple types of cilia. So there are motile cilia, which I've just shown you, which have a characteristic appearance of the 9 doublets and then a central doublet pair as well. And this is in contrast to primary cilia or sensory cilia, where you lack the central doublet. And in general, the primary cilia then are not motile, and they're located one per cell as opposed to the motile cilia, which are clustered in bunches. And then finally, there's this unique category of embryonic nodal cilia, which are the ones that are important for left-right patterning. And they're characterized by being one per cell, but being motile. So in terms of diseases of the primary cilia, these are all genetic syndromes known as ciliopathies, which have low penetrance of cardiac defects as part of them. So somewhere on the order of 5% to 15% of individuals with any of these disorders shown here will have a cardiovascular malformation. And what's shown here in the colors, all of the ones in yellow are genetic syndromes caused by disorders of primary cilia that primarily have a renal presentation. And then in blue, Alström syndrome and Bardet-Biedl have different presentations, but obesity is a common feature for them. In red are skeletal dysplasias or skeletal disorders as ciliopathies. And then Joubert and those in that bubble have primarily a central nervous system manifestation. So quite a wide variety of syndromic disorders that you can get. And there will be some low rate of situs abnormalities and or cardiovascular malformations in these conditions. This is just sort of summarized in this review where you can see that most of these have some prevalence of left-right asymmetry defects. So then the next category is the motile cilia, which I've already given the case of, and there's this known overlap between PCD and heterotaxy. And the first paper on this was in Circulation in 2007. And at that time, they identified in their PCD cohort that 6.3% actually had heterotaxy instead of normal situs or situs inversus totalis. And then a follow-up study done in 2014 showed that at least 12.1%. The reverse study asking how many patients with heterotaxy have primary ciliary dyskinesia has not really been done. But this definitely has management implications for patients with heterotaxy if they would have PCD as their underlying cause. So there's medical and surgical management of cardiac malformations, and then there's all of the diagnostic imaging that needs to be done for heterotaxy. But with PCD, you would add on those additional things that I talked about in the case related to the chest physiotherapy, periodic assessment of sputum cultures, and so forth. So in terms of clinical genetic testing for heterotaxy, there's a number of different options. So chromosome microarray analysis should be a baseline because of the non-random association with aneuploidies and copy number variants. I think testing for SIC3 should definitely be part of any evaluation, although somewhere between 1% and 5% of newborn males with heterotaxy will have a pathogenic variant in SIC3 that completely alters the recurrence risk counseling. So if it were positive, the recurrence risk would be 50% in males, where if it's negative, the empiric recurrence risk would be around 5%. So those are important numbers for families to have. I think testing for PCD genes and heterotaxy is very important because of the management implications related to that if we found it as an underlying cause. And then if there is something unusual about the presentation, considering testing for ciliopathy genes would be important. So I was never fond of really doing exome testing entirely for heterotaxy unless you had really good CMB calling as part of that, or if you did both exome and CMA. Now that we have the availability to do genome, that's really my test of choice for these patients. So this is an old paper, but there really hasn't been good updates in terms of molecular testing and what the yield is. I will tell you that from the patients that we've seen over my 10 years at IU, I think our yield is much lower than it probably should be for molecular testing. So sometimes I worry that the clinical interpretation part of it, that not all of the genes that might actually be positive are being classified and interpreted on the reports. There are data, though, on copy number variation and heterotaxy spectrum disorder. So if you just look at CHD patients across the board, the numbers are sort of 3 to 25% with extracardiac abnormalities and 3 to 10% with CHD patients with isolated heart defects. And then if you look specifically at heterotaxy, there's a couple papers that look at this. The numbers are pretty similar in these two different papers, and around 20 to 26% that will have a diagnostic CMB. So then just sort of as a summary with regard to the genetics of heterotaxy, clinically we have CMA availability, we have molecular testing availability, and then, there's research testing. And so on microarray, probably 15% on the lower end is more accurate, and then clinical sequencing, 10 to 20%. And so there's still a lot of room to go with regard to disease associations from a research testing standpoint. So for the last bit of time here, I'm going to talk about some of the research-based exome that we've done looking at these patients. So this is a little bit of a mixed cohort of laterality probands. So more than half of them have classic heterotaxy, which we define as a classic cardiovascular malformation consistent with heterotaxy, as well as situs anomaly in at least one other organ system. There's situs inversus totalis, and then isolated congenital heart defects that would be within the heterotaxy spectrum. So we did exome sequencing on 279. And then the first thing we wanted to ask is just on a research basis, if we look at the clinical panels that are currently out there and we do clinical grade interpretation on these exomes, what's our yield going to be? So we made a compilation of the clinically available gene panels that were out there, came up with 225 genes that we were going to look at and interpret clinically. And so our prioritization and sort of pipeline to really look for Mendelian causes, the main reason to show this is just that we set different thresholds for an X-linked inheritance model versus autosomal recessive versus autosomal dominant, because autosomal dominant, you get so many potential variants back. So we set a very stringent filtering thresholds, so they had to have a CAD score greater than 40 for us to retain that variant, along with some of the other bioinformatic prediction findings. So this is probably a minimum estimate of what we would consider clinically relevant findings in this cohort. So this is what we found. So we found 23.7% with positive results in this extended laterality panel genes. And so that compared to a 10% diagnosis rate if you just used a heterotaxy panel. So not surprising, if you add more genes, you're going to get better yield. But it does show that the heterotaxy panels that are available are probably not really the best test to be using in these patients. We did find evidence of X-linked heterotaxy in almost 4% of the participants. Actually, not only ZIC3, but this gene BCOR, which causes a syndrome that has been described, but has not been described with laterality defects. So that's an area of ongoing investigation for us. And then almost 10% with autosomal recessive disease, including about half of those with primary ciliary dyskinesia, and then 3.6% with a syndromic ciliopathy, including Bardet-Biedl syndrome, that had, kind of been missed and just enrolled as a sort of regular situs abnormality patient. So this is a summary then of the causation identified by research exome. And so overall, we had, this includes actually the CMV findings as well. So overall, we had 5.7% that had a CMV that was diagnostic, 22.6% that had a single nucleotide variant or indel that was diagnostic, and then 1.1% that had a dual diagnosis. And then, our two cohorts that are subcohorts that are probably big enough to say something about with confidence would be just the classic heterotaxy, which was very similar, and then our isolated congenital heart defect group. And the isolated congenital heart defect group had a slightly higher amount of CMVs found in it. The other thing that was interesting about that is that we did have some families that had a family history, either of heterotaxy, that was a little less common, or of first-degree relatives with congenital heart defects. And surprisingly, in those familial cases, we still were not very good at identifying a diagnosis, only 31%. So that indicates that we really have a lot of opportunities still in terms of being able to identify additional causes. So then the next thing we wanted to do was to try to identify other genes that were associated with heterotaxy or causing heterotaxy. And of course, we did the whole exome analysis first, but there's just too much noise there to be able to get something back. So we narrowed it to candidate genes, discovery genes for burden analysis. And the hypothesis here is that there may not be single gene causation, but you inherit an increased burden of variants in genes that are important for either the cilia function, planar cell polarity, or this TGF-beta signaling pathway. So it's sort of looking for multiple hits within a developmental pathway. So 967 candidate discovery genes. Again, had a variant filtering strategies. We did about a 3-to-1 matching of our European heterotaxy probands and then 1,000 genome controls. And so because this was a different bioinformatic pipeline, the first thing that we did was just sort of a sanity check. We had our patients that we had given a diagnosis to, a Mendelian diagnosis. And so we took that cohort out, and they should have had an increased representation of findings compared to 1,000 genomes controls. And that's what you see here with the p-value of 0.008, the 1,000 genome controls versus our positive, diagnosed part of the cohort. And then with the undiagnosed part of the cohort, there was no significant difference, which is what we expected because those were the individuals that we didn't find anything on with our previous analyses. So then we looked at sort of 296 genes from the Cilia database. And interestingly, we find additional hits within this undiagnosed cohort. And then there is a separate database, a Cilia database gene. And again, we find a significant difference between cases and controls with regard to that cohort that was undiagnosed. So, this tells us that there definitely is a differential inheritance, differential burden of rare variants in our cases versus controls. And then I'm not showing it today, but we've looked at another subcohort where we looked at the individuals that had classic heterotaxy versus the individuals that had isolated congenital heart defects. And what we see is that we maintain this statistical significance between cases and controls for the classic heterotaxy cohort, but we lose that significance for the isolated CHD patients. So we think that we, within these groups, have identified genes that are really specifically affecting the patients that have classic heterotaxy and less so the patients that have isolated congenital heart defects within that heterotaxy spectrum. So I mentioned that we did CNV burden as well. And what we see here is that between the those that had a single nucleotide variant, those that had a CNV that was identified, and the cohort that was undiagnosed, that all of those are statistically significant between them. So it's not a case where very often a CNV and an SNV are explaining the diagnosis. And so this is a summary of the overall level of variant burden. And this is a fairly complicated slide, but what you see in each row going across is an individual gene, and the color marking is the number of individuals that have a variant in that gene. And so each column then is an individual patient. So only patients who had a finding are shown, patients who were negative are not shown on this figure. And so you can see the number of subjects that then have multiple rare variants, and that's just shown by these stacked bars at the top, that, you know, at least half of the cohort has inherited multiple rare variants. Also, the color coding, you know, tells you the type of variants, and you can see with the black squares are cases where there are multiple hits. So this sort of led to a question of, is digenic inheritance something that may be possible? We know that, for example, in Bardet-Biedl syndrome and some of the heritable arrhythmias, digenic inheritance has been an important facet. And so we did just an initial analysis of this, looking to see if there were any pairwise combinations that were overrepresented or more likely to occur than what you would expect by chance. And we did find, although our numbers are relatively small, what you can see here with the dark green squares are those with a significance value of less than 0.01 that were more likely to co-occur. So in summary, I've mentioned that heterotaxy results from abnormal axis development during embryonic life, and that has made it somewhat challenging to study by some of the newer technologies because of the difficulty with targeting precisely the right time point. Heterotaxy can be inherited as X-linked, autosomal recessive, or autosomal dominant, but most frequently it's polygenic or multifactorial. And I think the studies that I showed at the end are the beginning of a path forward to being able to understand more of the polygenic contributions. Clinical genetic testing is available, but can be improved. And just, you know, the overall research-based exome findings that we had when interpreting in a clinical context was close to 30% with positive genetic results. And I mentioned already that, you know, just from the heterotaxy patients that we're sending testing on clinically, we're not close to that 30% number. So we need to find out why that is and where we're missing that. And some of that may be being able, especially now that we're doing genome, being able to re-review those genomes and actually see if we can identify some misdiagnoses. I think importantly, with regard to the question of does genetics matter, you know, 6.5% had primary ciliary dyskinesia, 3.9% had a syndromic ciliopathy in this study, and X-linked inheritance was found in close to 4%, which has important recurrence risk implications. So Not surprisingly, perhaps, we know that cilia-related candidate genes contribute to increased burden, and I think the opportunity and challenge going forward will be to better understand that individual level burden. How many hits do you need to inherit in order to really develop heterotaxy or a laterality disorder? And with that, I will stop. Very thankful to my clinical collaborators who helped with all of this, and I'll take any questions.
Thank you for the wonderful talk. I do have a question, and sorry if I'm outdated. Is that caught my attention in that slide study that you presented of variant classification, that you have like the 267 BCFs, and then you filter to get some variants at the end. At some point in the selection process, you apply the CAT score as like a criteria. I'm very curious. Well, the question is, did you just rely on the CAT score, or did you use other in silico predictors? If you just use the CAT score, why just that one? I'm just curious about that. Thank you.
Yeah, so we've done it a couple different ways now. It's a good question in terms of like how to do the filtering. So we used multiple bioinformatic programs. So there were 7 that we used in terms of, so we filtered first based on allele frequency, and then filtered using bioinformatic to try to enrich for rare, potentially disease-causing, potentially pathogenic variants. And then, but it was hard to know how to set a cutoff in terms of the inheritance pattern if we wanted to model different inheritance patterns. So we used that CAD cutoff solely for that, for when we only had a single variant in a gene that would be predicted to have dominant inheritance. We required higher stringency from CAD, because otherwise there were just so many variants that we would have to look at that would have passed our filter. Does that make sense? Yeah. And then when we, I guess when we did the candidate gene analysis with a different bioinformatic pipeline, there we were using, it's essentially the same thing, but meta-SVM, which is a different way of kind of incorporating all of the bioinformatic predictions to try to help you identify potentially deleterious alleles.
So what is your approach to children who are not have congenital heart disease that prior to start?
Yeah, that's a great question. So the question was, what about older kids? What do you do if they haven't had genetic testing, or if they have had it, do you update the testing with newer methods? So we honestly don't, it's relatively infrequent that we get older kids referred to us. Sometimes if it's like a new diagnosis, and there, I still recommend doing genome. If they've had analysis previously, it depends a little bit based on the pedigree and exam and those types of things. I mean, in an older individual, you can usually, with a good exam, rule out the syndromic ciliopathies. By history and physical exam, you can determine whether PCD is, a concern or not. So that really leaves you kind of with the known genes that cause isolated heterotaxy, which are going to be low yield via testing. So I err on the side of making sure that they have the recurrence risk information that they're going to need for those. I mean, I think a lot of times these heterotaxy panels that you send, or sending exome, what you're going to get back is a lot of variants of uncertain significance. And in an older individual, I'm usually more concerned about making sure that they have the clear known disease causing diagnostic findings that would help them from a recurrence risk standpoint. Like I said, if you've been able to rule out PCD and other syndromic ciliopathies. All right, thank you.
Thank you for that wonderful talk. For our last talk of the morning, we will hear from Amy Shikany. She's a genetic counselor 3 for the Heart Institute at Cincinnati Children's Hospital. She earned her undergraduate degree in biology at the George Washington University and her master's degree in human medical genetics at Sarah Lawrence. We will be hearing from Amy today about genetic testing for congenital heart disease.
All right, thank you so much for having me speak. And it's hard to follow all the amazing speakers that have already gone. I'm really going to focus on integrating clinical genetic testing for congenital heart disease, specifically focusing on infants and adults. So as our prior speakers have already detailed, there are 2 main different types of genetic testing that we need to think about, and both categories are super important when it comes to congenital heart disease. The first category is chromosome testing, which Joe reviewed, includes microarray, FISH, and karyotype, and the second category is gene sequencing, which can include anything from familial variant testing, single gene analysis, panel testing, exome, all the way up to genome sequencing. And we utilize all of these things for congenital heart disease. This is the current breakdown of the etiology of congenital heart disease based on our knowledge. About 15 to 25% is thought to be chromosomal, 10% is thought to be single gene variants, about 10% is considered to be teratogenic, things like maternal diabetes. But the vast majority of congenital heart disease remains unknown, multifactorial, multi-genetic factors contributing. When we look at neonates with congenital heart disease, about 20 to 30% of infants will have an extracardiac anomaly, so something outside the heart that counts as a second birth defect. And that is the category of individuals that we have the highest suspicion for an underlying genetic etiology. So in 2007, the American Heart Association came out with reasons for why genetic testing should be considered for congenital heart disease. Their reasons include because there could be other organ systems that need to be looked at. Genetic testing might provide important prognostic information for clinical outcomes. There may be important reproductive risks that are important to the parents and families. And then there might be genetic testing or screening cascade type analysis that needs to be done in other living at-risk family members. And the scientific statement was then updated in 2018 to further support the use of genetic testing in congenital heart disease, as the ability to do genetic testing was expanded with things like next generation sequencing, really acknowledging the high variability of genetic forms of congenital heart disease and reduced penetrance. But there are more reasons to do genetic counseling and testing in individuals with congenital heart disease. We know that there's high personal utility for patients and families. They like getting genetic counseling, and they have important things that they take away from doing genetic testing. And even if genetic testing comes back negative or normal, while not completely ruling out a genetic etiology, it does sometimes allow for lower risk stratification. So sometimes a lower recurrence risk. Maybe it means we're not dealing with something like an autosomal dominant condition, and so we rely more on the empiric recurrence risk. It also likely puts the patient in a likely lower risk category for complications associated with genetic syndromic disease. So in 2010, the consensus guidelines were released in the American Journal of Human Genetics that recommended doing chromosome microarray testing as a first-tier clinical diagnostic test for any individuals with developmental disabilities or congenital anomalies. And so this included individuals with congenital heart disease. And at Cincinnati Children's Hospital, where I am, this largely drove how we implemented clinical genetic testing specifically for infants with congenital heart disease. So first, we're going to focus on infants, and then we're going to focus on adults. When we think about infants, a couple considerations to keep in mind. About 70% of congenital heart disease is diagnosed prenatally. So we know prior to birth that this anomaly has occurred. And we never really know if a congenital heart disease is isolated or if there's going to be a secondary finding in a fetus, in a neonate, in a baby, or if they're going to develop a secondary feature at some point. And so I'm going to go through the process for how we integrated genetic testing into our infants with congenital heart disease. This process first began in 2010 with Dr. Ware when she was still at Cincinnati Children's Hospital. In phase 1, all babies that were admitted to the CICU for congenital heart disease received a postnatal genetics consult. So this included both infants with isolated congenital heart disease as well as infants that had known multiple congenital anomalies. And the genetic evaluation either included a genetic counseling encounter or a genetic counseling encounter plus an evaluation with a clinical geneticist. And I will show you how we decided when that was utilized. The majority of the testing utilized in this phase was microarray, but it was largely driven by the patient's phenotype and the geneticist recommendations. And so our findings from this initial phase were published in 2020 in the Journal of Pediatrics, and I will share some of that with you. So this is the algorithm that we utilized to determine how genetics would be involved with infants with congenital heart disease. Essentially, if a baby had a non-isolated cardiac defect, then a genetics consult was placed, a genetics physician came and did a dysmorphology exam, and they decided what testing was most clinically appropriate, whether it was rapid FISH, routine chromosomes, microarray, or gene sequencing. However, if the individual had an isolated cardiac defect, most of the time they met with a genetic counselor like myself to review genetic associations, talk about family history, review empiric recurrence risk data, and then we gave the family the option of doing a chromosome microarray. If the chromosome microarray was abnormal, then again, they would go back to see the clinical geneticist. And so using this strategy over a 5-year period, we found that the overall diagnostic yield was 26%. And this included both isolated heart disease as well as multiple congenital anomalies. Through this experience, we found that more than 30% of infants were having 2 or more genetic tests. So it's not great when we're having to go through multiple cycles of testing and it's somewhat redundant. As I had said before, the most common test that we utilized during this time period was chromosome microarray. But super interestingly, 17 or 11% of the infants that were identified to have isolated congenital heart disease had an abnormality found through gene sequencing as opposed to microarray. And so this really highlighted to us that we needed to be thinking about gene sequencing in individuals with isolated heart disease as opposed to just offering chromosome microarray as the first and only test. So then there was an updated consensus statement that was released by the European Heart Rhythm Association in association with the Heart Rhythm Society, and they further provided guidance in how to use genetic testing with special considerations acknowledging that there is both sporadic, syndromic, and isolated congenital heart disease. And this is a table from that publication. So they provide projected diagnostic yields of chromosome microarray, WES is whole exome sequencing, and WGS is whole genome sequencing based on whether the patient is syndromic, which means they have heart disease plus another finding, whether they're non-syndromic or inherited, could be multiple family members affected, or whether they're just the sporadic case in the family and no other findings. So looking at this category, both categories of testing are important and finding things within each of the different categories of congenital heart disease. Furthermore, this publication called out that neonates and infants with complex congenital heart disease are different than older individuals. And so they strongly support doing chromosome microarray or copy number variant analysis, but then also say that more extensive genetic testing could be considered. So things like doing targeted congenital heart disease gene sequencing, or thinking about doing trio, exome, or genome studies. And so after our initial 5-year period of collecting data, we decided that we had to adapt our algorithm so that we weren't missing some of these babies with isolated congenital heart disease that most likely probably had sequence variants that weren't being identified. And so phase 2 was initiated in October of 2020. And there was another important change that we made in this phase, which was collaboration with our fetal heart program, because 70% of our infants were diagnosed before birth. And so in this phase, prenatal or postnatal genetic counseling was occurring. And this is specific to infants with isolated congenital heart disease. So we no longer had a geneticist embedded in cardiology. And so the rest of this experience really tracks the course for infants with isolated disease, because if there was a baby born who had multiple congenital anomalies or was super suspicious for a genetic syndrome, we referred them on to human genetics so they could determine the proper phenotype and recommend testing that was phenotype driven. The genetic testing that we primarily utilized in this phase was a congenital heart disease gene panel that was done on an exome and then a genome backbone that allowed us to get copy number variant analysis data, so one test. The goals of making some of these changes were to get an earlier genetic diagnosis, because testing could be initiated at the time of admission if we had already met with the families, provided counseling, and got consent. We also really wanted to improve the family experience. The time to meet with a genetic counselor is much better outside of the CICU setting. Sometimes it's hard to track down mom. The parents are stressed. The baby is headed to surgery. And so we really wanted to meet with these families at a time when they could be more thoughtful and really more engaged in the genetic counseling process, as well as have continuity of care from the fetal diagnosis setting all the way into the CICU. And then we also wanted to minimize the cost and number of tests that were being sent. So we wanted to streamline and provide a more comprehensive approach to testing. So this is the updated algorithm that we utilized in phase 2. So you can see that isolated cardiac diseases still went through this algorithm, whereas if there was concern for an extra cardiac feature, IUGR, small for gestational age, dysmorphic features, they still went to human genetics. But if the individual had a prenatal diagnosis, then we had our fetal heart program referring those moms for a telehealth genetic counseling visit prior to delivery. And the purpose of this visit, well, sometimes, yes, we would talk about NIPT or other types of prenatal testing. Really, the purpose of the test was to the counseling was to focus on the baby's congenital heart disease and testing that would be recommended after birth. If individuals were not caught before they were born, whether it was a postnatal diagnosis or they didn't make it to genetic counseling for whatever reason, we would still meet with those individuals in the CICU at admission. And both pathways were offered the same genetic testing, which was a congenital heart disease gene panel on an exome or genome backbone. And so we were getting both the sequencing and the CNV data. And then we would refer to genetics based on results. So this algorithm was really a way of leveraging genetic counselors to practice at the top of their scope, while also knowing that geneticist availability was a problem at our institution. And so making sure that we were getting the patients that really need to see genetics prioritized to see genetics. So just a little bit more details about how we collaborated with the fetal heart program. The fetal cardiologist would refer for a telehealth genetic counseling visit. We would meet with them. We would talk about natural history and etiology of congenital heart disease. We would review family history. We would discuss different testing, including timing, obtain consent, and then document the plan. Once the baby was born and admitted to the CICU, a consult automatically gets placed. And so the genetic counselors would be notified. We would document in the baby's chart what was discussed from the mom's visit, and we would go ahead and initiate testing. When the results came back, obviously we would follow up with the care team and the family and make sure that everybody knew what the results were and what the resulting recommendations were. Partway through this study, we realized that doing the genetic counseling in a prenatal setting was just resulting in much more positive patient and family interactions. And so that prompted us to get a genetic counseling student to investigate the uptake of genetic testing for infants with congenital heart disease based on whether genetic counseling was done prenatally or in a postnatal setting. And this manuscript is currently in preparation. But essentially what we did is we looked at infants that were admitted to the CICU between January 2019 and April 2023 for isolated congenital heart disease. The study included 228 infants, of which 76 received prenatal genetic counseling via telehealth and 152 received postnatal genetic counseling in the CICU. And what we found confirmed kind of what we were already seeing, that patients receiving prenatal genetic counseling were 4 times more likely to consent for postnatal genetic testing than those that were seen in the postnatal period. We also did a little bit deeper dive into phase 2 outside of that student study, and we wanted to look at the yield of our testing using this algorithm. And so between October 2020 and July 2024, we saw 218 patients through this algorithm, and the yield of testing was 8.7%. 42% of those positive results were a single nucleotide variation, so we saw a range of things. We saw variants in Notch1 and elastin, and then we saw things that are associated with syndromic single gene changes. We also saw common copy number variants, things like deletion 22q and deletions of whole genes like Notch2 and SMAD2. The problem with this is that we were getting uncertain results almost half the time. So that high VUS rate was coming back. And so this is tricky when you're getting a variant of uncertain significance in an infant who you have limited history on. So trying to tease out what is suspicious versus what isn't, trying to test the parents in a timely fashion to see, is this inherited, is it de novo, is somewhat tricky and stressful for parents that are already in the CICU setting. So we then initiated phase 3 of integrating genetic testing for congenital heart disease into the CICU, which included everything in phase 2, except we changed the type of genetic testing that we were offering. And so as of July 1st of this year, we started offering genome, rapid genome with results in 7 to 10 days. Ideally, we do it by trio, but if that's not an option, we'll do it by duo or proband only. One of the big wins in figuring out the clinical flow and logistics of this process was we got one of the genetic testing labs to work with us so that when we meet with mom prenatally via telehealth and the family consents to do postnatal genome, we mail the parents buccal swab kits so they can swab their cheeks, send it back to the lab prior to delivery. The parental samples are already at the lab waiting. So when the baby's born and hits the CICU, we can send off a sample and it really expedites the results. So I don't actually have any data to share with you from our phase 3 yet, but I will give you a few examples of how this change in implementation has been super helpful by sharing several case examples. So this particular patient was a 29-year-old pregnant woman that was referred by the Fetal Heart Program. The baby had complex congenital heart disease that included a DOR, VSD, and coarctation. Prior to meeting with a cardiovascular genetic counselor, the patient already had an amniocentesis completed that included aneuploidy fish and a reflex to a SNP microarray, which was normal. The family was counseled and consented to genome sequencing with postnatal sample collection. We do offer secondary findings in terms of whether or not the families want these. I initially thought that that would be a difficult discussion to have with families prenatally, but it's turned out to be really streamlined. Most people are super open to it and have a strong opinion one way or the other. In this particular situation, they declined secondary findings. The family history was non-contributory, really nothing else going on. So the clinical course of this case, the baby was born at 39 weeks and 2 days. Testing was set on day of life 1. Head ultrasound was normal. Kidney ultrasound, the kidneys were small but otherwise normal. We do screening head and renal ultrasounds on all our infants admitted to the CICU with congenital heart disease, just to screen those body systems and make sure that there's not a second birth defect there. There was a delayed alpha diagnosis. So when we got the genome sequencing results back, this is what we found. We found that mom and dad both carried a pathogenic variant in NAD-SYN1. And so this is associated with multiple anomaly spectrum disorder, and it's really rare. It affects the heart, kidneys, and spine, and it's newly described. The gene is not even curated in ClinGen yet. There have been approximately 19 individuals reported. All individuals are known to have spine involvement. A large majority have congenital heart disease, including others with DORV, like our patient. About 50% have genitourinary renal anomalies, such as missing kidneys, and then there have been reported limb involvement as well as rib differences. The outcomes of individuals with this particular gene are largely related to the individual's structural anomalies. And so in this particular case, the results obviously changed how we were going to care and treat this family moving forward. So again, the renal ultrasound was essentially normal, but then we had the question of, do we need to do additional spine imaging? We actually reviewed the CT of the heart that was done on day of life 1, and we saw that there were multiple vertebral and rib segmentation anomalies that were identified. It just wasn't the most pressing issue at the time the baby was seen, and so it wasn't something that we noted until after the genetic testing came back. Obviously, because this is an autosomal recessive disorder, it informs inheritance and family planning, 25% risk for future siblings to be affected and 50% risk for future siblings to be a carrier. The baby's future children will be carriers. We know that spine and heart defects are super common but variable. And we didn't discuss reproductive options such as plantation genetic testing, but we could in the future. Importantly, there's no risk for developmental delay or intellectual disability for this particular disorder as far as we know at this point. And we were able to get this kiddo routed to see genetics in an appropriate follow-up time of 9 to 12 months. Here is case example number 2. So this was also a prenatal referral for genetic counseling. The patient didn't schedule, and that happens sometimes. So a postnatal consult was placed on day of life 1 for congenital heart disease. The baby had tetralogy of Fallot, polyvalvular dysplasia, and pulmonary atresia. Pre-test counseling and parental sample collection occurred on day of life 2. As you can see from the family history, the proband had a 2-year-old sister. Mom and dad were healthy, really nothing else going on in the family. We got results back on this kiddo, and they had a de novo or new partial deletion of CHD7, which is associated with CHARGE syndrome. Obviously, we know that CHARGE syndrome is a syndromic disorder that has multiple medical and follow-up recommendations, including they needed to meet with genetics, they needed ophthalmology, immunology, auditory evaluation prior to discharge, thinking about endocrinology, evaluations, brain MRI, as well as other screening. And so Do I think that the diagnosis of Charge syndrome would have been identified at some point if we hadn't had this genetic testing? Yes, they would have identified other features in this baby, but having the genetic testing and knowing that the deletion was de novo really was gave us the opportunity to optimize intervention for this baby. Okay, so now we're going to skip all the way through childhood and teenage years into adulthood. So a couple of considerations when thinking about genetic testing for adults with congenital heart disease. Approximately 90% of patients with congenital heart disease were born after 1990 reach adulthood. So there are 1.3 million adults living with congenital heart disease. Many of these adults did not have access, or the genetic testing was not available that's currently being offered to infants and children today. And so they're likely underdiagnosed. But adults give us more information in that they have more history to kind of guide testing approach, which can be super helpful when we're thinking about phenotype-driven testing. The European Journal, or the European Society of Cardiology released this publication, I believe it was in 2022, which outlined the cornerstones of genetic testing for adults with congenital heart disease. So similar to what the AHA recommended in infants and pediatrics, they say that genetic testing should be considered to confirm a diagnosis, guide management, inform long-term prognostic information, inform recurrence risk, and as well as genetic risk assessment in family members. They also say when should this testing or counseling be done in the adult setting. They recommend in transition clinic, preconception, or really for any older adult patients who have not had access to previous counseling. They encourage that all types of genetic testing be encouraged, but it also be done in the context of genetic counseling. This is the algorithm that came out of that same guideline, which is obviously somewhat complicated. And I don't know about you, but my adult cardiology colleagues were not super thrilled when this algorithm came out as a streamlined way to determine which of their patients needed to see me versus have genetic testing. And so what we did at Cincinnati Children's Hospital was to do a deep dive into our own clinical population first to really see what we were dealing with. So we had a genetic counseling student that did a retrospective chart review to look at the prevalence of genetic diagnosis, testing, and referral patterns in our adult population with congenital heart disease. She was also trying to compare the prevalence of genetic diagnoses in adults versus pediatric patients with congenital heart disease, and identify characteristics associated with genetic diagnosis, testing, and referral. We are hoping that getting this information would help us create a more informed algorithm and know kind of how to best intervene or what to tackle first within our adult congenital heart disease population. This was, as I said, a retrospective chart review. Individuals had to be more than 18 at the time of their most recent adult congenital heart disease visit, and they had to have congenital heart disease. So here are some of the findings from her study. You can see that in table 1, the breakdown of cardiac lesions. This was really broken down more so based on physiology than how we look at some of our infant cohorts, looking more at like the Botto criteria. We also collected whether or not there were acquired or congenital comorbidities, as well as a family history. If you look in table 2, you can see that the prevalence of a genetic diagnosis in this cohort was 13.7%. 13.7% of the cohort had at least one genetic test, and 11.6% had been referred to genetics. So the vast majority of these patients were not having genetic evaluation or testing, despite having a high number of comorbidities. When we looked at the different types of comorbidities in table 3, you can see that some comorbidities, congenital comorbidities, were more associated with a referral to genetics, such as craniofacial abnormalities, skeletal limb, or GU-IU features as well. We also saw that there were a high number of individuals with ADHD or developmental delay as well. And then the pie chart on the bottom shows the breakdown of genetic diagnoses by type. Probably not surprising, the majority had Down syndrome and 22q, which are obviously pretty common recognizable syndromes. So what did we conclude from this study? We saw that adults with congenital heart disease have genetic diagnoses at a rate comparable to those in pediatric populations. However, they're not receiving the same level of care because so many had not seen genetics. We think that all adults with congenital heart disease who have non-cardiac congenital anomalies or neurodevelopmental comorbidities or a family history of congenital heart disease should be referred for a formal genetics assessment. And adults with congenital heart disease with a history of limited genetic testing should seek evaluation and potentially be offered more comprehensive genetic testing when appropriate. So after we concluded that study, we wanted to do a second study to look at what institutions, what outside institutions were doing in comparison to us. So what we did was we sent out a survey to all adult congenital heart disease providers to assess how they were offering genetic testing and genetic evaluations to their adults with congenital heart disease. And we wanted to see what indications and practice structures influenced decision-making by the adult congenital cardiologist when it came to the provision of genetics care. So this was done by sending out a recruitment email. We sent an email to 113 adult cardiology providers through their professional society. We asked targeted questions. It was mostly closed-ended and some Likert type scales. The inclusion criteria was that they currently had to be practicing and a board-certified cardiologist seeing adults. So we had a response rate of 35, which is not awesome. It's a response rate of 34%, but I don't know if you've ever tried to survey cardiologists. That's actually a pretty good response rate. Some of the things we found is that the majority of individuals were referring between 1% and 5% of their patient population genetic services, whether that was offering genetic testing or referring them on to genetics. There was only one participant that did not refer at all to genetics in the last year. We also gave the cardiologist a couple of different clinical scenarios and asked them whether or not they would refer to a genetics provider, which I found interesting. So we asked them if there was a 30-year-old male with an interrupted aortic arch type B, would they refer? 74% said yes. A 16-year-old male with a VSD and developmental delay, 66% said yes. A 20-year-old female with heterotaxy and complex CHD, 43% said yes. And a 41-year-old with transposition and a family history of an ASD, only 40% said yes. So just interesting that obviously the higher associations with things like a known syndrome obviously increase the referral rate. And then if we look at testing practices, we saw that the vast majority of testing that was being ordered by adult congenital cardiologists was chromosome testing. So FISH, microarray, karyotype. There were only a few individuals that were sending any form of gene sequencing, which is really interesting because in adults, if they don't have developmental delay or maybe they don't have other things that are super concerning for an aneuploidy, we probably need to prioritize gene sequencing. And so just something to think about. The majority of individuals did report barriers to referring to genetics. And so there are definitely things that can be done to help smooth the pathway of genetics care between cardiology and genetics. In terms of influential factors, we saw that more years of practice tended towards less referral and less testing. We thought this was maybe because older adult congenital cardiologists maybe weren't educated in the testing technology since they were relatively new. We also thought that more testing was associated, or that seeing more patients would be associated with more testing, and it was. We thought that maybe this was because more resources are available if the clinic has an overall higher volume. We also found that greater confidence in recognizing syndromic features and greater confidence in ordering testing were associated with ordering more testing, which obviously makes sense. If they feel comfortable, they're going to do it. Something that was not an influence was evaluating pediatric patients as well. So we thought that if an adult congenital provider was also seeing pediatric patients, they might be more in touch with kind of genetic testing practices, and that didn't seem to make a difference. One of the things that came from this study was really that potentially adult congenital heart disease training programs should include more education on the inclusion of genetics. Right now, the adult congenital heart disease board certification has an exam with 2% of it covering genetic syndromes and associations. So there's lots of inroads we can make into education. So conclusions that came from this study: cardiologists are referring patients to genetics. Most of the testing ordering by adult cardiologists was for aneuploidies and microdeletions, when obviously we might need to be thinking more about single gene variants, and that there are many things we can do to improve training and access. So with all of that said, we have not developed a standing algorithm for our adults with congenital heart disease to come to genetics, but we have encouraged our adult congenital heart disease specialists to refer anybody who has an extra cardiac feature, history of neurodevelopmental delay, a family history, has questions about prenatal or preconception questions, to come to see us. And we definitely have had more patients come in. So I'm going to share a few of those cases with you. Case number 1 is a 23-year-old pregnant woman with a history of a PDA that was repaired and a newer diagnosis of aortic root dilation. She had been seen previously by genetics due to coarse features and concern for a connective tissue disorder. At the time, she had analysis of TGF-beta 1 and 2 and MYH11, which were what were kind of clinically available at the time she was seen. She had normal growth and development at the time that we saw her. She was in college. Here is her family history. So she was referred because she was pregnant and interested in recurrence risk. At the time, we didn't know anything about her fetus. She hadn't had a fetal echocardiogram. There was no other family history of anything suspicious. So the first thing we did was we ordered an aortopathy gene panel, and the results initially came back with a variant of uncertain significance in the gene collagen 3A1, which is the gene associated with vascular EDS. This didn't really fit the patient's phenotype, and so we weren't highly suspicious, and then we weren't surprised when this variant was downgraded to likely benign several years later. And so then the daughter of this individual was born, and they also had a moderate PDA, which closed spontaneously. They had a small anterior muscular VSD, a small atriopulmonary collateral vessel, and a PFO. The baby was also noted to have excessive hair growth, which sparked concern for CAN2 syndrome, and we ordered analysis of ABCC9, which is the gene associated with CAN2 syndrome. We found a pathogenic variant, which confirmed that the baby and the mom also have CAN2 syndrome. CAN2 syndrome is autosomal dominant. It's associated with coarse facial features, excessive hair growth, and skeletal changes. There is a high risk for cardiac features, including PDA, cardiac enlargement, dilated aortic root, and vascular tortuosity. So identifying this finding not only changed kind of the care for this individual's daughter as she grows up. We're going to monitor her aortic root. We're going to look for other skeletal changes. But with our patient, we were able to identify that she had vascular tortuosity and had some other skeletal changes that needed to be monitored as well. So it really had clinical importance for not only the daughter, but the patient as well. Here is a second case to share with you. So this is an 18-year-old who was again pregnant. So you can see that our adult cardiologists like to refer people when they're pregnant as a point of a good time to think about genetics. This individual had a history of tetralogy of Fallot that was repaired in the neonatal period. She had been tested for deletion 22q syndrome, which was negative. She had graduated from high school and had started trade school, so normal growth and development. And her fetus was identified, not changed on its own, to have truncus arteriosus. The pregnancy had non-invasive prenatal testing, which included 22q and was low risk. And so we were able to think about doing genetic testing in this family. You can see that beyond the mom, who is the proband in this case, there is a paternal uncle with arterial disease, but really nothing else of significance. And so when we got back this individual's report, this was when we did a comprehensive congenital heart disease panel, so looking at 400 syndromic and isolated sequence variants as well as copy number variant, and we found a deletion of notch 1. So going full circle here. We know that deletions of notch 1 are known to cause non-syndromic congenital heart disease, and so this explains not only the patient's history, but her child's, as well as informs recurrence risk. She is already planning another pregnancy and is using this information to help make decisions. So I kind of cheated a little bit and only shared cases with you where we found informative genetic testing that was positive. But in the majority of cases of congenital heart disease, genetic testing is going to be negative because we know most congenital heart disease is multifactorial or multigenetic. And so if testing is negative, we really fall back on providing genetic counseling for these patients. We talk about empiric recurrence risk based on the type of lesion that is present in the family and who is affected. We might make lesion-specific screening recommendations for family members. For example, if it's a left ventricular outflow tract obstruction anomaly, we provide psychosocial support. And then obviously if genetic testing was lower, as I said, or was negative, the recurrence risk is likely lower than 50% because we're not dealing with any of these common, well-known autosomal dominant disorders, as well as a likely lower recurrence risk for medical complications associated with genetic syndromic disease. The other thing I would just like to point out, especially with our infant population, is that we know infants are going to grow and change and have other findings. And so if genetic testing, particularly in that group, comes back negative or normal, we really emphasize follow-up, contacting us if there are any changes, a new concern in an organ system, concern with developmental delay, things like that. It really is kind of a fluid, ongoing process, as is interpretation of genetic testing. So thank you, and I guess my slides are done. Yeah.
How are you?
Good, how are you?
In your phase 2 on the pediatric, what was the barrier, or why did you think about just sending to is this because of barriers of sending those in the prenatal setting?
So we definitely do. So the question was sending prenatal genome, right? Okay. So there definitely are some individuals that have prenatal genome sequencing done, but typically our fetal heart patients do not want invasive testing. And so that is really the barrier for why that is not being done. Most of the time, they don't have objection to doing genetic testing after the baby is born, but they don't want to kind of take on the added risk with an amniocentesis on a pregnancy that already has complications and is stressed. So that is primarily why we don't do that. I would imagine that in other geographic regions, that's more common, but not in Cincinnati. Yeah.
Yeah, I think we have actually a pretty high genetic testing here. One question that I had really, and the adult data is really fascinating. It's clearly, and because so many of them were babies before we had genetic testing, so there are many, many adults who need the genetic testing now. But thinking back to where you talked about for neonates and sending the whole genome, how has your system addressed the cost and payer situation with that? Because that's a challenge all
cost?
Yep. So the question is cost for sending testing. And I'm going to answer the question in terms of thinking about more broad testing like genome. Is that fair? Okay. So testing in terms of genome sequencing up until very recently was somewhat tricky. Our institution does allow us to send genome sequencing for anybody who's inpatient. So there's no barrier if the individual is an inpatient. The concern for us was the outpatient setting. However, there is at least one clinical genetics lab in the last few months that will now not balance bill for Trio Genome if you have Medicaid, and they work with commercial insurance for some insurances, not all, but like United, Cigna, some of the big payers. So it's become much less of an issue, and the cost just has not been an issue. Any question? Yeah.
I was curious, based off that, when exactly do you go larger, like exome genome versus just doing a smaller, say, CHD?
So I think in infants, I would always lean towards going more broad, like genome or exome, because we have limited history. We don't know what they're going to develop. I think the cases where I might stay more targeted to a congenital heart disease gene panel is in somebody who is older and we know all they have is heart disease and nothing else. Any questions? All right.
All right, thank you.
All right, I know everybody's been looking forward to lunch. Let's return to our seats by 12:40 and we'll get started with the afternoon. Lunch will be served on the second floor ballroom and they'll be out there directing us down there. Okay.